And good morning, everybody. We're broadcasting live from the ROV Falkor in the Coral Sea. This is dive number 385 on the ROV Falkor, the ROV SuBastian, and we are currently
anywhere in particular
taking a core just right next to that. Thanks, J-Rod, it'll be perfect. You've caught us underway this morning on our first dive. This is dive number 11 of our voyage, number 385 in the sequence for the Falkor. We are currently on the very start of our morning transect. We're located in the upper reaches of a submarine canyon located offshore from the Great Barrier Reef. We're within a kilometre of the edge of the outer Great Barrier Reef system. Yesterday, those who are online would have watched us do a transect which climbed a very steep head wall of a canyon which sits right adjacent to one of the reef systems. Today we're looking at a bit of a contrast to what we saw yesterday. We're still in a canyon, but we're running a canyon up a canyon which isn't blocked by one of the reefs of the GBR. Instead, it has an open connection to the waters and sediments of the Great Barrier Reef. We won't be climbing as steep a rocky head wall, we don't think, based on our bathymetry mapping from recent days. But we do know we'll be going up through quite a variation in water depth. So we're starting at water depth at the moment of 517 metres. As you can see down in the muddy seabed at these depths, and we'll be tracking up the axis of the canyon wall, relatively low gradient, to a water depth towards the end of around 60 metres, where we should be, or we will be getting into the sunlit waters of the upper end of the canyon. So we've just taken 2 short push cores with the SuBastian. This is part of our standardised sampling methodology we use on all of our transects. We take duplicate cores to give us sufficient material to work with, and we'll bring those back on board towards at the end of the dive and process them in the lab here on board the ROV Falkor. SuBastian's now taken off, so we're now starting our transect, running along in a direction which will take us in a broad south, heading toward the southwest direction from where we've started. Looking at the seabed in view, it's quite steeply sloping. Just getting a sense of the pitch and roll of the SuBastian right now, though. We'll just come around. So the seabed is dipping across our view into the murky depths. Little fish darting across there. So very much like the start of yesterday's dive, where we're seeing quite barren, soft sediment seabed areas, occasional fish. We're flying a little high at the moment, so we'll certainly aim to drop down shortly. The weather on topside today is another beautiful day on the Great Barrier Reef, here in the Great Barrier Reef Marine Park, where we're authorised to come into the park as part of our voyage to make these observations of the canyons and collect samples. We're focusing in on what we call the epibenthos, the life forms that live on the seabed. But of course, any other observations we make of some of the in-fauna, such as the crustacea, the worms, we also record those, but we're not sampling those in particular. Just as I speak of those, we're now starting to get into a really nice view of again what we saw yesterday, these wavy lines across the seabed, which quite possibly, quite likely in many cases have a biological origin in that they're formed by organisms, worms, crustacea crawling across the seabed, leaving their traces behind. There's a burrow, quite large burrows we're seeing quite commonly in these areas. Water temperatures down here, 9.3 degrees Celsius at present.
You can see it.
We can zoom into that.
See it's having trouble going up the hill.
Yeah, it's quite steep, so it's crawling up.
This might be why we're not seeing so much life, on this face.
Yeah.
Right? It's hard for animals to even climb up it.
Right, yeah.
Without any hard substrate, it's vertical.
Welcome to our broadcast this morning, Jeremy Horowitz, who's on board the Falkor for our entire cruise. One of our resident biological experts. Jeremy's very keen on the hard corals and the black corals.
Hard corals, not so much.
Okay. So the start of our transits after waypoint 2, is that right? Okay.
Yeah, it's just on the bottom.
Okay. Bit of squat lobster? Or, what have we got there? Some sort of spider? Some sort of spider? Any ideas? Got 8 legs, yeah, good call. Partly buried.
See through his
Yeah.
head.
Oh, okay.
There's somebody in this hole right here.
And then down there, just in the bottom of the view, there's looks like a little fish.
There's also what's that up to the right, that black thing, Jeremy?
To the right?
To the right of the spider.
Oh, is that a little fragment of coral?
Let me zoom out.
Do you want to zoom in?
Oh, this bit? Okay. Gradually, carefully zoom into that.
I imagine it's a fragment of
Something-
what could be a black coral that is either, it's falling down this muddy slope slowly, probably.
Yeah.
You would have a touch on it, make sure it's not connected.
Yeah, why not? Give it a waft. I mean, it seems like it's just kind of hanging
Loose,
right through.
yeah, not ideal for sampling.
But that might mean
that's the
there's
case.
something shallower. It doesn't look very heavy.
Falkor is just manoeuvring its
Small.
manipulator arm to this tiny fragment of coral. We're just trying to work out whether it's loose or whether it's anchored in vaguely anchored. No, we'll call that loose, shall we, Jeremy? Yeah. We'll leave that alone. We really only want to collect material that's in its what we call in situ habitat location, you might say. Okay, we'll carry on.
All right.
When the seabed's as bare as this, your attention gets drawn to the smallest thing. But there's an interesting, following that little trail.
Is that a pond?
It looks like a fish, but
Definitely looks like a fish.
What are those flatfish?
Yeah, okay. I'll zoom right in to see its colours. And his eyes, look at that. That's pretty neat.
Yeah, it would be like a flounder, right?
Flounder of some sort, I'm hearing.
The deep fins.
Forward facing eyes, not side facing.
Cool.
Good.
Okay, nice.
Just for the hell of it.
Just for the hell of it. Ah ha ha.
You're on a roll today, buddy.
We've got a few comedians in the control room this morning.
I was missing 2 legs.
This is good. Okay, yeah. A legless crab.
Yeah. Yeah, can you give me a call?
Right, still. We'll probably see this for some time. The seabed, more or less barren of epibenthos. Occasional burrow into the seabed, and then these myriad of wavy lines, which are a record of the activity of the infauna crawling across and shallow depths within the seabed to disturb it, regularly turning it over, oxygenating the sediment. And we see this in our cores. When we take a short core, we see an oxygenated top layer of about 5 to 10 centimetres thick. So it's all pretty consistent. Couple of characters. Shrimp. Small fish. So just to put people in context again, we're coming down water depth of 525 metres. Currently tracking down the lower edge of part of the canyon wall, and we'll hit the canyon axis and then track up the other side, is the basic plan. And that'll take us then climbing up towards water depths of 60 metres. Timeline today, at least 5 hours on this, we expect.
What's going on?
Perhaps longer, depending on how we go in the shallow waters. Yeah.
One of those rat fish.
Bit of disturbance. Interesting behaviour, whether that's a... There's one guy in action flying across, kicking the legs.
You wouldn't think they're good swimmers.
Yeah, you wonder about their direction control. Not great. Okay, we'll fly on. Definitely a lot of activity this morning on the seabed with the fish, crustaceans. Yeah. How's the current, guys? Just out of interest.
Not bad.
Not bad current. Okay. They took their hands off the controls there for a moment to see how ROV SuBastian might have moved, but it didn't. So. Pretty similar to yesterday. Didn't have much of a current either then, which was good. There's some sort of a sea snake in the back.
Hey Leo, when Angus fills up the hot tub, can he fill it up with
Worms and stuff?
We're just zooming in on this chunk on the seafloor. Don't know if it's rock or wood. Remember, we're only about 50 kilometres from the coast here. Yeah, that's wood, and it's got little gastropods all over it. Yeah, these are a lot of snails. What do you think, Jeremy? Is this worth getting? It's interesting. I mean, on beaches you can walk around and the wood washes up, obviously. It floats in out of the Pacific and out of rivers and the like. Don't often see them down on the seafloor, though. This is kind of interesting.
Why don't we suck up?
Yeah, let's have a.
Maybe we could get a sense of what animals are actually attached to the wood.
Compare with the ones that break down the bones. I don't know if you'll have your colleagues that would want it.
Don't worry, I'm not taking over. It's just some photos.
I mean, maybe at
Ooh, there's a crab.
how big is it?
It is
That big.
Oh, okay, so we could put it in a box 34.
So in the tropics, when pieces of wood is very easily colonised by an animal called a teredo worm, and they're a real problem
30 cents.
in the 1800s. With ships were mostly made of wood, and they tried all kinds of ways to stop these teredo worms biofouling or chewing into the wood. In fact, the teredo worm is not a worm at all. It's not a polychaete. It's actually a gastropod. So it's a mollusk, and they have these. They bore into wood, and they have a calcareous casing, and so all those tunnels that you see inside wood is actually not made by a worm at all, but made by a gastropod. So here we have a bit of wood sitting on the seafloor. I'm not seeing any teredo worm holes, but we're seeing a lot of the your classic gastropod snail. Oh, this is a squat lobster sitting underneath. Sorry to disturb you. It's actually not a big piece of wood. It's about 10-20 centimetres across. And there's a lovely shrimp underneath. Beautiful colours. And we're just going to have a bit of a look at what we've got growing here. So we've got a couple of squat lobsters and some urchins on the underside. I'm seeing some amphipods or isopods. There's quite a bit of life on this piece of wood here. And it isn't hugely burrowed. But if this stays down there for much longer, of course, it will be completely eaten away. Lots of life on this. We got some smaller crustaceans. I'm not going to say that they're squat lobsters, or they might be. Yeah, the tails are actually bent in underneath. I think that's what we're seeing here. Lots of smaller ones. But of interest too are these tiny little white gastropod snails that are attached to the outside, and they don't appear to be burrowing, but just simply attached to the outside. but they could be just feeding on the organic matter that is made up by the wood itself. So that's really quite interesting. There's a real great little habitat here. Just a tiny bit of wood sitting out on the seafloor. It may have just been simply washed out from the rivers nearby. We're only 50 kilometres from the coast, and of course there's lots of rivers and things along there, and we have rainforest right up close to the edge of the Great Barrier Reef in this latitude here. So, you know, this wood could simply have been washed out of the rivers and dumped onto the seafloor as soon as it got waterlogged. Lots of life there, yeah. And if you have a look on the left-hand side, there's a sort of tube-like shape. This is a classic teredo worm casing. Now, I'm not saying the animal itself is inside it, but even after it's dead, the teredo worm can leave these classic long white tubes behind. And, you know, in the old 1800s, they used to line the sides of their ships with lead. You know, actually, sheets of lead it served two things. It formed a ballast to keep the weight down, but it was also to protect the wood of the sailing ships from this biofouling, because it used to just eat it away. You know, and it was mainly an issue in the tropics. Of course, nowadays we have ships of steel, and we use all kinds of biofouling chemicals to try and stop barnacles and the like, because, you know, with that biofouling, it really slows down. It causes lots of friction and stops, you know, it slows ships down. So it's far more efficient to be able to stop the animals attaching in the first place. Yeah, this is a lovely squat lobster on the other side. Bit happy that we're playing around with his home. Yeah. So yeah, great find, guys.
I imagine it's a really good thing to hold on to.
And we're looking at the urchin. There was a couple of urchins on the underside of that piece of wood.
Right back.
And we'll just plonk it straight back. Sorry for disturbing your morning there, guys. And they're all heading back into their home. We've got this lovely shrimp that's just beautiful colours. It'll go and settle itself back in.
Thanks, go.
Okay. Hand you back to Scott now. Thank you.
Oh, yeah. All good. On we'll go. Push on. Right, back to the featureless seafloor, and these biotubation mounds coming into view. Oh, here we go, Jeremy. Here's something in the lower left. Something, one of your... have a close look at this.
Surely. Is that soft coral?
Didn't zoom.
Might be worth collecting. Interesting, you know, the few corals that do survive in this kind of habitat. Yeah. Oh, beautiful. Yeah, let's take a...
We'll take that.
So are you...
I got it.
You got it.
Yeah. Look down a little bit.
Move down a little. The other way. The other way.
Oh, yeah. Fair enough.
You're manual a little bit.
There we go.
Ah, beautiful.
Get some good images of this before we take a sample. You got the lasers on?
I gotta look down and zoom out a little bit.
Okay, right.
They're like down there.
They're down there, okay. Right.
I want to zoom out a little bit.
This is
Okay.
wood, okay.
There you go.
Okay.
Can we see wood?
All right. Actually, let me just quickly take a look at the way it's. Just okay.
Okay.
Yeah. Throw it in.
No research.
I guess 1A. Or do you want to leave those open for runs? Yeah. Oh, absolutely. Put it in box 2, please.
Box 2, after that.
They go on something like contour parallel. It's just too. What did you see? Did you see a couple
saw-
of contours?
Yeah.
Equinox.
What was it that we saw right in the beginning? It was trying to climb up and it kept sliding down. It was.
It was an urchin, was it?
It wasn't an urchin. It was like a. No, it was a crustacean.
See what this is anchored
Couldn't
to?
climb up. It kept trying, and so you imagine, that's how you go up or down something. You go.
If anything.
Follow these deep parts, don't you?
Yeah, he's deeply anchored, isn't he? That's
You want to.
guys having to.
Okay. That makes sense.
Okay, that's good. Good sediment sample with that too. Happy with that. Oh, look at this length of that root. Do we call that a root? Yeah, that is called a root in coral terminology.
Going back to how quickly the
Almost
sediment accumulates, that might have been living tissue at one time that got buried and died. Right?
Ah, yeah.
So it hasn't
Okay.
sent that down.
Interesting idea. Yeah.
Actually, yeah.
Yeah. Okay.
That looks like a sea pen.
Yeah, of course.
Nice.
Currents on there, but it was, eventually you can see somebody comes out of that. It's actually a source of organic matter, like this carbonate fibre of vegetation that comes out. So we should keep an eye out for organisms.
Just carefully laying that long specimen down into the bio box for later processing up on board in the labs. At the end of each day we turn off the live streaming, but then we've got a few hours work ahead of us still with taking all these samples we've collected during the dive and sorting them and preserving them, either generally in ethanol for the biological materials, sometimes a bit of formalin, depending what it is. And our geological samples, with the cores, we carefully handle those, extrude them from the core tube and transfer them into sample bags, straight into the fridge
You wonder what it was.
for later transport back to our labs in our various offices.
Or does it have that root system?
I can just do it. That means it can go anywhere it wants. It's got a root hold and stuff.
And this is pretty hard sediment.
Nice sea star.
What's this on the right? Is that
It's another stick, I think.
stick?
Another stick.
I think.
So we're starting.
Significant, we're seeing
Yeah.
logs and
Yeah, so-
vegetation in this canyon.
Exactly. Yeah.
So
So
if we go deeper,
yeah, that's just a bit of a stick, tree stick.
would we see a dead forest?
It looks a bit like a piece of mangrove.
That'd be cool. So as I said at the intro today, this canyon has an open connection to the Great Barrier Reef, lagoon waters, and to the coast beyond that. So it's not that surprising we're starting to see these evidence of terrestrial land input this far away from the coast. In these depths, I should say.
It makes sense.
It does make sense.
Fish going past.
Yeah.
I'm not so steep. You wouldn't go up, would you? You would go sideways.
It's a long way up, isn't it?
Getting a good perspective view here of the steepness of this part of the canyon wall as we fly along it. The ROV's flying flat, so that's a true view if you're diving down to these depths.
We're just coming up the southern sidewall.
Okay, so it is.
Canyon, so it is climbing up.
Yeah. And we're not far from the start of our 500 metre standard transect, so let's keep an eye on that on our tracking maps in the control room here. What have we got there? It's a little flat fish of some description.
Yeah.
There we go, look at that, he's got a... Wow. Amazing structure.
Yeah.
It's got this fins.
Semi-translucent.
It's got these pointed bosses.
Sensei fish.
A sea robin.
Let's just make that up.
That'd be a gurnard.
Yeah, good.
Sea robins are gurnard.
Okay. Any fish specialists out there, please? Oh, I've got a sea robin online. And here we got another large something living under it, a fish. Is this a bit of... Yeah, let's have a look at this, please.
Is this an outcrop?
So, yeah, this is the question. Or is it a bit of algal mat that might have...
Or just eroded timber. This looks like
Ice coral.
underneath it.
Yeah, there's something living in there. We don't want to be too rude.
It's like a net, a mesh of some kind.
Is this...
Amazing. And
There's something. There's a fish under it.
so this
Is
is...
this...
I'm not positive, but
Looks
I'm
like
pretty sure that's
organic.
There's a lot of debate going on here as to whether it's organic or...
Brittle stars.
Yeah, I reckon it's organic. Yeah.
Amazing.
Mainly
And there's a fish. We'll leave the...
Isn't that something?
And all over it, Jeremy, you've got little stars, worm tubes.
Yeah, lots of brittle stars. Yeah.
And there's a
Tubes.
perfect habitat for that little...
And, you know,
Harry's...
why there's a hole there. And you can,
That's the front door.
what a lucky fish.
Prime real estate in this environment.
There's no rescue mission. He has a hole. Get out.
That's right, yeah.
To rescue.
We'll leave them alone, of course.
Interesting. That's really cool.
Yeah, okay. Novel.
Definitely looks something organic.
Yeah. Is it something washed in off the reef?
Yeah.
Because the only source for that.
Yeah, it could be vegetation, like leaf, some large leaf or...
Yeah, okay.
Got a lot of stuff living on it.
Really something.
Yeah. Really goes to show, once you have a bit of complex, harder substrate, life kicks off in these environments. there's clearly hundreds, if not several hundred organisms living on this thing.
Just poking his head out.
Yeah, he's saying, "What do you guys want?" I can't eat.
I can't see.
You're being streamed worldwide, my friend, on YouTube and Facebook.
Awesome.
Let's excavate a nice little burrow underneath.
Okay, let's move on to our 500 metre transect.
That was very cool.
That was interesting. Sort of just.
Like a big mat.
That was about 30 or 40 centimetres across.
Could be.
Yeah.
Nice upon burrow, maybe.
Is anyone home? No, no one home.
No one home.
No one home.
Do you know how long it takes an isopod to make a hole?
No, I don't know. But did you see it scooting over the seafloors? I mean, it had little jets. It's a little digging machine. Underwater version of a wombat.
That's right, that's our tag for them on this leg. Wombat holes under the water.
Nice, good sized shrimp over here. Good size, good 10 centimetres. Just put another gurnard. More of these.
All of these burrows.
Something was in that one.
Yeah.
This was the depth that we saw the isopods in yesterday. So pretty reasonable to think that these are isopods too. Lots of excavation.
Yeah. That's right, lots of biotobacco.
Burrows,
trails, yeah, that's right.
Again, a fairly large urchin.
Yeah.
The mound that's coming up in front of us here.
And then,
Oh, something to the left.
there's a crab.
Was it a crab or something else?
No.
More left.
More left, there you've got a sea pen.
I want to just zoom in. We don't need to stop. We would just look at it.
Okay.
Go ahead.
It's not. Oops.
It's like another sea pen. Yeah, looks similar to what we just collected. I don't know if it was the same red color, but that umbellium, I think it is. Yeah, similar.
Okay. No collection there, we're happy.
That's okay.
Right, we'll move on to our 500 metre transect.
Almost at the start.
Yeah, okay.
All set.
So stand by for start of 500 metre transect.
Ready.
Okay, we're on. Thanks, Alex. Thank you. So we've just commenced one of the standard things we do on these dives. We include a 500 metre transect, we call it, or sub-transect of our overall transect, that gives us a standardised view of the seafloor as we fly along. We don't sample along this section. Instead, we fly at a constant height, and in the control room here we have a team, including Alex Post and Steph McLennan, both from Geoscience Australia, who are capturing images as we go, still images, representative shots of the seafloor, using specialised software called Squiddle. And that allows us then to tag and get the precise locations of the things we see along the way, and it includes not only the biota but also the substrate type, as in muddy sand in this case, or sandy mud as the case may be. There's more wood there, Alex, in the background.
Happy with that speed?
How's that speed, Alex, for you?
Or a little bit slower.
Okay.
Tad slower.
Slowing it down.
Thank you.
Are you seeing too much in every shot?
Is that better?
Yeah, that's great. Thank you. That's a good speed. And you can tell from the angle of the seabed here, we're starting to ascend. So water depth currently 495, and that'll continue to reduce.
Is that a straight shot, or are you happy with that?
Yeah. Alex, do you have a preference for the angle?
What's that, sorry?
Perpendicular.
It's flying a little.
Yeah, I mean, that's going to be sort of the nature of this transit.
Yeah.
We are sort of going a little up.
Yeah, it's a bit of bleak. We'll stick with this. Thanks, Cody. Yeah. Something large looming in the... We've got our first rock for the day, maybe, folks?
Well, you said we never stopped during the transects, but that's not exactly true.
True.
Yeah, except
If it's-
Dumbo octopus.
That's right.
We stopped the Dumbo octopus.
Right.
We try not to
and
stop.
Nautilus.
Nautilus and Octopus.
Yeah. We can slow down and let's have a look at this. So this is interesting. We're in 490 metres.
Look at these.
We've
the
got a
sea
major
turtles.
block. I wonder. Is it
Soft coral there.
sort of carefully fly over it and get a sense of the size of this thing?
Little stars.
Wow.
Exactly.
Okay, this is continuing up.
With black, with Gorgonia.
Gosh, it's huge.
Yeah, it's very
This is a big block.
But it's interesting that it's still pretty bare. Like,
Yeah,
you
okay.
can imagine this is an area where all the corals would survive in this area. Maybe it's raining down too much.
Is this a debris flow, Rob?
Was it Gorgonia?
Is it reef-like? It looks
is this a
reef-like.
debris-
It's got
is
lots
it-
of cavities in it.
So are these loose blocks,
Debris,
debris flow kind of idea?
Yeah.
Yeah.
We found blocks at this depth on the canyon yesterday.
We did.
Oh, isopod, big isopod,
Yep. They're
Giant
crawling out of
isopod.
its.
just in front of us, up the top of the screen. Big.
So
This is probably Bathynomus. Look at that. It's good size. The beauty. Oh, this is what we think is creating these large burrows that are in the
Possibly.
muddy seafloor. Oh, isn't that beautiful? Yeah.
So looking at the rock in the background too, there's the clear
It has that limestone
reefy look to
about
it.
doesn't
Yeah. There's a burrow.
We could save your pause in the transect.
Yeah.
It's
As long as we
but
sort of
very
get that,
ancient looking animal, isn't it? Like you could.
as long as we sort of zoom out again to get
Yeah.
that consistent view.
Yeah.
I can just
We'll go back to where we
analyse
can.
those images.
Yeah. Okay.
Yep. Just let Brandon know.
And,
You could almost, think of this as like fossil,
Trilobite.
hundreds of millions of years old, fossilized. It has that really ancient look about it.
Like a trilobite. Did you say that?
Yeah.
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 beautiful, nice sponge on there.
So this is on our 500 metre transect. We haven't taken a detour. This is just a fortunate thing, but we've zoomed in, which we wouldn't normally do, but for obvious reasons, to see the detail,
Add it to the list of things.
and then we'll zoom out again and re-establish our constant height, which is very important.
This crab or lobster's actually got some sponge or something on that, on the upper, on its carapace. Lots of cup corals. Yeah, this is definitely reefal.
Reefal,
Yeah.
limestone. There's no the way
Limestone,
that
but
open
probably not in situ. not
open
at this
framework.
depth.
No, this is a block or a block field. I'm
Barnacles in the
saying.
background. Just here.
Oh yeah.
If they're
Oh yeah.
decent sized lobster. And look, there's lobsters all over it. Do you see these?
Yeah.
The antennae sticking out like they're all over it.
There's a whole colony here, really, isn't it? Yes, that's the point.
Can we zoom in on the sponges here? Just like right there.
If it's a world first.
Unusual shape.
They each
Almost
have those
glass.
lobes.
Yeah.
They have those.
Yeah.
Should we grab one?
Your call.
Yeah, let's. Is it how? Can you get there?
Yeah.
Yeah, pick one of those two.
Take one. Yep. So we've just broken our rules, but we'll do, these are the
There too.
opportunities you can't miss,
Yeah.
really. So
Got look, having a bit of a battle on the.
on the side there, we've got
we got Neil and
we got
crayfish.
Oops, we'll
That looks like a glass sponge.
disturb the piece there.
It's small. It's maybe 5 centimeters across. So glass sponges are one of the target groups that we're collecting on this expedition. I think I'll take this opportunity to explain why we're collecting corals and sponges on this trip. In addition to understanding what makes up the Great Barrier Reef, we also are interested in understanding what lives within the Great Barrier Reef and the Coral Sea. And the reason being, it's the fundamental information that we use to protect biodiversity in these habitats. So if we don't collect these specimens and identify the species, we can't say for sure what they are. And the only way to know for sure is to collect them. And we now have these advanced molecular techniques that allow us to identify the species that we're seeing and compare it to other closely related species that one might think are the same, but could be geographically separated and therefore potentially molecularly different. And so we have collaborators. We have a sponge collaborator. We have Merrick Ekins at the Queensland Museum. Yeah, how about we skip this one and maybe look down. There was another one. There was one more, right? Yeah. But it might be.
Trying to knock one off.
Yeah.
Yeah. Grab it after that.
Yeah. So these are glass sponges. They're made of pure silica. So they're pretty hard. So when the manipulator arms get a hold of it, it tends to crack. Like if you were to squeeze a piece of glass, it wouldn't have much give.
Right.
You think you could scrape it off at the base and not?
Trying to do, but
Yeah.
I touch it, so it's just falling apart.
Yeah.
Okay.
Right next to you.
So we have J-Rod trying to delicately remove the sponge from the substrate. And you can see how small it is. So it's a delicate procedure. Yeah, we've collected a few glass sponges. I would say one of each type that we've come across. And we haven't collected one that looks like this yet. Oh, yeah. Maybe.
Bit hard, eh?
I can't, now that I got it loose from the outside.
Yeah, it's hard work, that one, isn't it?
Yeah, if it's
Jay,
proving too difficult, that's okay. We'll find a better opportunity.
We'll move on, I think. Yeah.
The only other thing we could do, Jeremy, is try to suck some of this up. It's not going to be a lot of
No. Well,
Don't worry. We'll get there.
sometimes it doesn't work.
Okay.
Okay, we'll zoom back out and resume our 500 metre transect.
Thanks for trying, J-Rod.
Yeah, thanks guys for the effort.
Relation. You want me to give it a like? Yes.
Sure.
Just looking online, we have someone signing in from Southampton at 1:45 in the morning. Well done, David. Great to see the dedication and interest.
Is this what you wanted right
So,
here? Did you want this kind of overview? Right here or more back down?
we're just going to reposition ourselves back to our transect line properly.
right. It's settle a little bit.
Settle here.
Settle.
No, settle.
Just letting the dust settle. Water column settle.
Yep.
And then resume.
Resume.
That's it.
Yep.
Very good.
It's important we have as clear a water column as we
How's
can
that
get.
one right there?
It's a bit of a closer look than that, but yeah.
A little bit closer.
A little closer.
So this is an outcrop. This is a rock outcrop.
It is a.
It's more than outcrop.
The interesting thing, we saw this similar rock at the same depth yesterday in our other canyon head. So, and that time we were pretty confident it was in place.
Yep.
So I'm
That's
a
right.
little hesitant to call it at the moment as to whether this is in place outcrop or a debris flow from higher up. It's just we haven't seen quite enough of the context to really understand. These are definitely blocky, and each of them one that is a sort of a scale which is quite plausible have come down as part of a landslide flow, but
Okay.
yeah, for me at least, the jury's out a little bit on that right now.
Alright, back on transect.
But we'll piece the jigsaw together over time and come up with a bit more certainty. There's another rock, obviously, in the
It's the same stuff.
same stuff. Yeah,
It
thanks.
was a really big outcrop that went up somewhere.
Yeah, thanks, J-Rod.
It went up quite high as well.
Okay. Our ROV pilot's just mentioning in the background that looking at the forward-looking sonar which we have equipped on the ROV SuBastian, they've seen this outcrop extending a long way up. Now, the ROV the sonar is used to keep an eye out for in the distance any hazards that might be coming up, as in large rocks or caves that we don't want to fly into. Can happen. What have we got here? Is this more of this matte
That cool?
organic? Yeah. Dead coral. Is that the tubes?
Sponge?
That's brittle star.
Wait, living on it? Yeah.
That sponge.
Thick.
Like sponge, yeah.
Again, we'll call that debris that's come down from higher up.
Yeah, I'll let you know if it gets put up. So we're tracking along at 483 metres depth. Gentle ascent at the moment across the canyon floor. Constant height above the seabed, part of our 500 metre transect. Fish. Water temperature at these depths currently 10.4, so obviously we're more warming the waters gently as we come up from the depths. We started today at about 4 or 5 degrees from memory.
Shall we put the sonar up on the live stream again?
Ah, thanks, guys.
The rock outcrop.
Yeah, very good. That's great. So just on the live stream, you're seeing everyone. There's the sonar signal, and that black zone on the left-hand side, sort of the left quadrant, is part of a rock outcrop. So it creates a shadow effect on the sonar screen. So yeah, we'll have another block of rock coming for us to see, sort of starting to come into view now. Okay.
It might be worth pausing the transect depending on what we see.
Yeah, no, this is looking more outcroppy. It's extending back up slope. You can see, and it's a big piece as well.
Bottle.
And another bottle we should tidy up.
All right.
We need to do a bit of cleaning up. 2 bottles.
Pause the
Oh, yeah.
transect and just explore this rock.
Yeah, good call, Jeremy. We'll do that.
Are you keen to clean up the sea?
Yeah, we'll collect the bottle. Be irresponsible not to. We are in a World Heritage Area after all, Great Barrier Reef Marine Park. So we'll do a little bit.
So we're seeing cup corals, lots of cup corals up here.
Yeah,
Tusk shells. Curious to know what kind of bottle it is.
Looks like a beer bottle. This is Australia.
Goes with the Great Ball that we found yesterday.
Yeah. Is that a bit of plastic?
It looks like a bit of plastic, and which is
Accumulation zone here.
Oh, there's another good one.
Another what?
Joe, is that a bottle down on the lower left?
Yeah.
Yeah. Yeah, thanks, mate. What's inside the bottle? Someone's asking. Yes, a message in a bottle. Who knows?
Nice.
What box can we put it at?
Which box?
3 or 4?
Sure, it's empty.
Yeah, man. Ooh. Okay.
Oh, that was a shame. Wow. Yeah, the guys didn't squeeze it. It just shattered. It's pretty old, I guess. We've still got a lot of it. Hopefully we'll be able to pick up that broken piece, which is sitting in the... Yeah, we'll tidy it up. Ay.
Alright. I don't think the depth, not much of a depth change from where it was to where it is now. And it's open, so.
Oh, you're still safe.
This is around the same depth.
Someone commenting on our turtle again. Only one strapped on today. Yeah.
Curious. It's a glow-in-the-dark turtle. So
This one is a...
if we turn off our lights,
Do you want to turn them off while we're... Oh, maybe not.
Yeah,
At this
turn 'em off
moment.
yeah, we can give it a shot.
Yeah, for sure. Yeah. Oh, no.
Can we see if this turtle goes underwater? Alright, here we go. Lights are going off.
Alright, so this is a glow-in-the-dark turtle, so we're going to see
If it does.
if it glows in the dark.
It's
turning off lights.
Ooh.
Maybe turn off the lasers too? Turn off there.
Don't worry, folks, your screen hasn't gone black. Well, it has, but we haven't lost signal. We're just doing a
Yeah.
bit of a test of the glow in the dark. It's coming on now.
You want to find zoom focus? Oh, there it is. Wow, look at that.
So, it glows. Hooray.
That's cool.
That's awesome.
Experiment.
Experiment complete. All in the name of science.
That's right. Thank you, guys. Those turtles are made in a 3D printer on board here. Great design. Flapping
All right.
flippers and everything.
Are you going to pick up the other bottle?
Back to the transit.
So, I think we'll take a real quick look around this rock, just see if there's anything of interest before we resume the transect.
Oh, there's another bottle. Okay. Gosh. Yeah.
All right, attempt number 2 on grabbing a bottle with a metal arm. I did need something to put more ethanol.
Yes.
Good container.
Reuse, recycle.
That's right. You don't have to pump quite as often, right? You could
Yeah.
have ethanol in there and aliquot it.
Is that technically upcycling to do?
I'd call this upcycling.
There's something living in it too. Comes from the sun.
All right. So we're gonna take a quick spin around this rock. It's an outcrop before we resume our transect.
Quick look at it.
Yeah.
You want to
I'll-
zoom in here?
Yeah, I'll just pan a little bit left and right. Is that all right? Okay, we got cup corals. Some dead, some alive. Mollusks, shrimps. Or cup coral.
Just turn it back to sand.
Crustaceans up top.
Not just breaking it. No, don't break that one.
Fly to the other side?
Sorry.
You want to fly to the other side?
Yeah, in 1 second. Oh, hermit crab. Yeah, lots of cup corals. A little hermit crab on the cup coral.
Combo is there. Yeah, it's on. Like I had a loose grip. You said I had to do yesterday.
Brittle star
I was like, used to do a crazy thing like
coming into view.
randomly flip out for
That's pretty cool.
just really getting crazy striving at night. Just to kind of
Yeah, we could spin around. Yeah.
name this a big fun one.
Science came out for you, squared.
They're on is everything on the AC. AC, I mean,
Majority.
that's why I flipped it around. So that's a 50 days.
Yeah, so the camera, well, that's what we see. And your camera is shutters. Right.
That's the big not the same. Thanks to the show.
The road for shell. Another shell. Would you like me to flip it over?
That's what science is all about, isn't it?
It's under.
We're curious to know what's underneath this really large shell. Also curious to know what the shell is. Nice.
Something's wrong with
It's a mellow.
Put it back.
Yeah, sure. I'll just, yeah, it looks like we're all right here. Yeah, look at resume. I just see cup corals and,
Can you turn tech?
yeah.
So we're doing the translation.
Yeah, we'll get back to our starting point.
Yeah.
Or resume. We call it a checkpoint, is that right?
current stuff and they collect this really big gold and
Alright, you ready?
yeah.
Okay, good beginning transit.
Alright, we're just resuming our transect now.
Lasers back on. I got it.
I can see my mother's watching. Hello, Mom.
Hi, Jeremy's mom.
You gotta wave, Jeremy.
Wait, your mom.
Wave to your mum.
I think my mum's on watching as well, so. And my neighbours.
Yeah.
Good speed?
Yep.
Awesome.
Scott Nichol back in the seat. Just took a quick coffee break after that rock. So back now, water depth 466 metres. Continuing our transect, 500 metre transect. Gradually climbing the wall of this submarine canyon. Seeing these occasional rock outcrops, which are all good to see. But overall, it's still a sediment-draped canyon floor. Slightly sandy mud, which will be coming in from a combination of just raining out from the water column, fine-grained particles, organic material, but for the most part, sediment coming in from the Great Barrier Reef lagoon itself, through the open entrance that this canyon has. Open connection to the reef. So with every day, every tide coming in and out, and then occasional larger energy events, river floods here in tropical North Queensland, of course. They're very common every year. Cyclones also kicking up sediment. These lines on the seafloor in places are very well defined, very sharp edges to them. So unquestionably they have some degree of biological origin, but I'm thinking that the gentle currents on the seafloor are also helping shape them a little bit, to gentle currents to remove the very fine particles, the silts and clays, and leave behind sand-sized particles, maybe up to 2 millimeters in size. And typically they're lighter in colour, whiter in colour, and they're standing out proudly on the seafloor as a distinct signature of that process.
$350 to go.
350 metres to go, thanks J-Rod, on our 500 transect, so we're not halfway yet. Total length of today's dive will be about 1 kilometre. So, some hours to go yet, which is great. I've got a whole day to look forward to of this. These are rare experiences for us as marine scientists and geoscientists. We plan these trips for, in this case, it's been at least a year in bringing this voyage on the Falkor into fruition. Navigating all the COVID challenges, of course, along the way. So we're extremely fortunate. Every day we get up and look at each other and say, can't believe we're here. You know, we're actually on this vessel undertaking these fantastic dives. And delivering on a plan that we, as I said, we started over a year ago thinking about. And Rob Beeman, as you heard in some of the other commentary, our colleague from James Cook University, has been working in this area for over 10 years. So for Rob, this is a really important step in that long-term research program that he has in this part of the Australian marine environment, where Rob has focused a lot on the Great Barrier Reef with his colleague Jodie Webster at the University of Sydney. So it's great to be part of that now from the team here at Geoscience Australia. We're part of the Commonwealth Government based in Canberra, and we have a team that undertakes seafloor mapping at a national scale, and we take opportunities like this to get involved in the research as well. Keeps us connected to the technology in a way that opportunities don't often come along, and we actually then make use of the data that we collect on these voyages into building up the national picture of what Australia's seafloor looks like in terms of its seabed geomorphology and composition, its evolution, and also importantly the habitats that go along with those seabed features, like canyons. So today we're in a canyon. It's one of those, for the Australian margin, quite a common geological feature over the Australian margin when you take a look at the big picture. In fact, we've mapped in excess of 700 canyons on the Australian margin right around. And this section of the northeast Queensland continental slope and shelf is one of the areas par excellence where we have some beautiful illustrations of canyon form, where we have the canyons cutting up into the continental slope and then incising into the edge of the continental shelf of Australia at a depth of around 100 metres at that point. But this is very much a collaborative voyage. Those of you who have seen the web page on the Schmidt Ocean Institute website will have seen the every voyage, every cruise has a web page dedicated to it, so you can go to that and read about some of the background of how we got here and why we're here, and all the collaborators involved. There's about 10 institutions all linked into making this happen. Very collaborative, and it's setting us up for many years into the future of working on the data, carefully piecing it together, which will lead to scientific publications, but also a lot of outreach as well, like we're doing right now, live streaming to the world through YouTube and Facebook, and publishing online blogs, and in time we'll be releasing other products from this voyage, which include things like bathymetry fly-throughs, where you can get a little movie of the seafloor, as you're seeing right now on screen. In fact, that's giving us a nice view of the canyon system that we're in today, with that little red line, as well, the one we're tracking along. But as you can tell, looking along, there are many other canyons along this margin, this little area we're working in. So we'll generate bathymetry fly-through movies just like that, which we tend to annotate and give a bit of context to.
1.8. Walking across the
Yes, Emily, hi there, Scott here. That's lovely, Bethy, as you say, smiley face. We're blown away with the bathymetry we were getting. It had been mapped before, so Rob had a 30-metre resolution bathymetry grid prior to our voyage, which was essential for planning. But now every evening we're remapping our study area, our sampling area, and fine-tuning, bringing into sharper focus the seabed features. So now we're using a bathymetry grid which is at about 10 metres resolution. So it's been really helpful to do this on-the-fly planning, which we have to do each day. So we'll come into the voyage with a broad plan of where we like to go and pretty clear ideas of where we want to position the ROV, but then we've been tweaking it just to make best use of this high detail bathymetry. So, for example, this morning we had a line plan ready to go and we realised we hadn't quite got it right position, so a few last minute waypoints were quickly punched into the system to give us the best opportunity to see the features on the seabed.
Did you change the Falkor monitor?
So we're maintaining this seafloor substrate type as we fly along on our 500 metre transect. Slightly sandy, mud dominated, bioturbated, barren seafloor. Barren in terms of the epibenthos for the most part, but there's unquestionably a lot of diversity, biodiversity, just in that shallow seafloor area with those worms, crustacea of various sizes. Oh, okay, here comes another one of these bits of debris, we think. Yeah, another bit of wood. Seen a few of these today. Again, not that surprising. Again, we've got an open connection to the Great Barrier Reef lagoon. And rubbish, as Robs says, yeah. Two more bottles to the collection today. Fish. What's that shiny silvery There's perhaps a small, yeah, I want to say sea snake, but I'm not sure. Maybe too deep. For 10 metres now. Water temperature 12.5 degrees, so it's kicked up 2 degrees since I last read it out for you all. So it's gradually warming. Yeah, thanks, Rob. Right on screen now is tomorrow's dive for those planning their next day's viewing. We'll be hitting a canyon tomorrow which has not incised its way up to the edge of the continental shelf. It's what we call a slope-confined canyon. Some people also call them blind canyons. Blind in the sense that they have a dead end. So that's tomorrow's action. We won't get too far ahead of ourselves quite yet. Crustacea. Is this a sea cucumber coming up, do we think, or another? Quite a lot of jellyfish now just coming into view too. Ctenophores. As part of this overall voyage, we are on some of our dives including a dedicated period of time to closely look at the jellyfish. We have a colleague in Japan, Dr. Dougal Lindsay. Not sure if you're online today, Dougal, but yeah, so we next planned plankton dive will be on Thursday of this week, Thursday morning. So those of you with a particular interest in the jellyfish, because they're brilliant and beautiful when we really slow down and look at them closely. But there's quite a few coming in right now. We'll be recording these for Dougal. We've got a couple of smaller cameras dedicated to recording that particular view into the water column. So Dougal will have an opportunity later on to identify the species floating past. Sea pen from this distance is what we'd call that little stick. There goes a shrimp. So things are getting steeper as we fly along. Here's my perception. And yeah, Chris is sitting next to me, the ROV pilot, is nodding. Yes, it's getting steeper. 400 metres. Yeah, okay, so thanks. Just on screen we've got that 3D perspective view of the bathymetry of the canyon. And you can see a yellow dot. That's the ROV location right now as we come up canyon. It's interesting, you know, on these steep slopes, in sediments, you get a thing called an angle of repose. That's the steepest angle that the sediment can pile itself up to before it collapses as an avalanche. Now, it's a bit challenging to measure that here and now, but we're approaching that angle of repose. But for mud, you know, it's much more cohesive, so you can actually stack it up a bit steeper in places. It's fine, mate, yeah. It's painting a bit of a picture. Thanks. Yeah, so just to make a slight correction there, we are flying obliquely along a canyon wall. It's where our liner is taking us, but nonetheless it is a steep slope. Three, just broken through the 400-metre barrier, as it were, climbing up. Okay, thanks. Just reading online, someone's done a quick online check for us to deepest water depth for sea snakes of 250 metres. So, okay, we're way beyond that. So I'll stand corrected there. So,
Do you think Ben can argue for his class for me?
certainly in my experience, I've only ever seen sea snakes on the sea surface or in the shallows from the safety of a boat. Here in Australia, every time you see a snake, you just assume it's venomous, regardless of where you are. That's right. Everything's venomous in Australia. Having a chat with the pilots. Perhaps a coral off to the left in the distance there, Jeremy. Vaguely making something out.
Are we still doing a transect?
Yeah, we're doing a transect, so we'll just take that as an observation.
Yeah, probably another CPEN.
CPEN, yeah, we've had but this is barren from an epibenthos perspective. Pretty obviously. Yes, question? Do we spend any time documenting wrecks or see any at all? Haven't seen any on this voyage, but the answer is yes, we do. As marine scientists, marine explorers, if you like, we do from time to time come across shipwrecks, and when we do, many of them are charted already in Australian waters, so there's a record of the location of shipwrecks, particularly those from World War II here in Australia. There are a lot of ships that went down, including here in the Coral Sea, of course. They are much further offshore from where we are now. But I do know the national vessel, the national research vessel for Australia, called the ROV Investigator, has in recent times, in the last 2 years or so, spent some dedicated time mapping shipwrecks, including off the coast of Victoria at the southern end of around Bass Strait. And if you were to, they've been really great in publicising those discoveries, so you could find images of those shipwrecks as seen in the multi-beam on their website. Just Google or put into your search engine of choice ROV Investigator shipwreck discoveries, and you'll probably find something. So marine archaeology is a very important area of activity, but we're not totally focused. Sure, if we found something we'd spend time, but that's not our objective. So, more jellyfish floating by, and it's becoming a bit more gentler in the slope as we track this bit. Yeah, okay, so we're sort of coming to a slightly flatter bit, but something coming up in view.
Tube anemone?
Yeah, thanks, Jeremy. Tube anemone. And there's a large fish just floated past higher up, didn't it? Yeah. Which reefs are you between, please? 5. Get my numbering right. Hey, Rob. Excuse me, Rob. Ribbon Reef 5 is just to the north of this.
Yes, Ribbon Reef 4.
Between 4 and 5.
I'll show you a picture quickly.
Yeah.
Ribbon Reef 5, Ribbon Reef 4.
Okay.
The gap between them is 2 kilometres. It's a 45 metre inter-reef channel.
Okay.
The 5 is 6.5 kilometres long. This is, I'd say, about 6 kilometres wide each, about 1 kilometre, sorry, long each, 1 kilometre wide each. 60 k's from the coast.
Get a chance. So that was a black coral to the left? Oh, we don't know. Jeremy's a little...
I'll let the transect just continue.
Yeah, hopefully so. So nothing... We just had a question on Facebook about which reefs we're between, because we are between 2 of the reefs. For those familiar with the numbering system, we're between Ribbon Reefs 4 and 5. And the gap between those, laterally, north and south, is 2 kilometres. So we're more or less coming up midway between those 4 and 5 reefs, halfway along that 2 kilometre gap. Another question about whether we're taking push cores. Yes. Oh, really? Just
Liking the spotlight.
liking that. Attracted by the light. Yeah, we took a push core at the start, down in 600 metres water depth. We'll take one effectively toward the end, or if we get a clear change in sediment texture like we had yesterday, where before we started to ascend the cliff wall, we had a lot of coarse debris that we could take a core into. So we'll try and replicate that. Thanks for that question, Luke. Great to see people interested in the sediment record, because that gives us a really important archive of environmental information and the processes operating on the seabed when we take these push cores. Even though the cores are quite short, it's still a really important insight. And it adds that overall context setting for understanding the habitats and the way they are responding to change, as we've just clearly kicked up a bit of sediment. Oh, okay, I'm hearing that was done by fish. Not us.
It wasn't us.
Someone's not happy we're here. Yeah. God, there's a lot of burrow traces there right now. This is interesting. 370 metres. You know, we've had biotubation all along, but I'm just, my eyes telling me we've just kicked up a notch in density in the last little while. More little mounds. I know my colleague at Geoscience Australia, Rachel Preslowski, if you're listening or hearing this later on, here's some beautiful infaunal burrows. As part of our work at Geoscience Australia, we work across a whole range of water depths from the deep abyss up to the shelf, and over time we've been building up this knowledge of the distributions of these sorts of habitats, in particular the infauna and the biological traces they leave into the sediment, because they get incorporated into the sediment record and into the geological record as well. I know Rachel's written a blog on the cruise webpage, which you can find on the Schmidt Ocean Institute website. Rachel's written a nice little blog talking about her experience in discovering these markers of biological activity on the seabed called Lebensperren. I know she'd be quite excited if she was here. She's part of the team for this voyage. Ooh, okay.
Looked like a shark from the shadow.
We got distracted by a fish on another camera, everyone. Sorry about that. It didn't broadcast that one. But oh, we did. Okay, cheers. Thanks, guys. It's hard to keep. I did a count the other day. In the control room, there are 32 monitors. So you can miss things. There's a lot of repeat vision, I've got to say, but there's a lot going on. Yes, as I was saying, go to the webpage for the Schmidt Ocean Institute, find the dedicated page for this particular cruise, which comes up first when you get there. Seamounts, reefs and canyons of the Coral Sea, and there's a growing list of blog stories which members of the science party, some who are onshore, some who are offshore, are contributing to the narrative for this voyage, relaying some of their personal experiences and their scientific motivations for being involved in this voyage, and from their careers as well. Always great reading.
Trying to get closer, but it's so much higher on the right side. Where are we ending? Or is it the end? It's halfway between the next two. Oh, we have two different ends. We have waypoint 8 and we have halfway between waypoint 7 and waypoint 8. Which one is it?
For the end?
And says it's halfway between waypoint 7 and waypoint 8. Over here, it's right on waypoint 8.
I'll check with Rachel, with Alex, who's just left. Okay.
It's not very much further to go all the way to 8. Do that one just to make sure we have coverage.
Yeah, double check. Yeah, we're running a bit longer. Thanks, guys. Okay, seeing a bit more coarse sediment now on the seabed. We're at 362 metres depth. So all those white flecks will be bits of shell, perhaps halometer flakes, which grow in the Great Barrier Reef lagoon and get washed down. Very light, platy, flat flakes, which like a thumbnail, toenail kind of size at biggest, and they float down. So this is to be expected as we get shallower and closer to the reef edge. We're seeing much more of a concentration of that material coming in from there through that gap between Ribbon Reef 4 and 5. There's a couple of I don't know my fish at all. Have we had an ID on the fish? No, we haven't.
I think they're amberjack fish.
Amberjack was a one-offer. Ah, that's nice.
That helps with their ID.
Just a question on Facebook as to how far into the water do we see our lights projecting from the ROV, and it's around 10 metres, so it's not a massive distance. But these are really high-powered lights. Remember, this is pitch black, of course, when we don't have any light. And the lighting's a critical part of the ROV success story. You can't just use any form of light. It's just that getting the right balance. So we project light in from all angles and to get an even view across the seabed. And then the technicians adjust the cameras to get the right white balance. So prior to a dive each day, we do a calibration check once we hit the seabed, so that we're seeing as close to true colour as we can. Yeah, some of the shells are now they're getting bigger and they're more or less intact. Cone shells, perhaps. Yeah, okay, so starting to see some of these shallower water shell species, just the remnants of them as well. Excuse me. That's good. I'll finish this one. Temperature's now risen to 14.1 degrees Celsius, so a couple of degrees warmer from only about 50 metres deeper, so yeah, progressively warmer as you'd expect. And there's a nice, again, inset view on screen of our track line as we climb the southern wall of the canyon valley. Okay, we're looking downhill in that view, just got my bearings. And yeah, the position of the ROV just coming into view now, thanks guys. So that's lovely panning back now, and you can see that greenish tilt area in the lower left has just gone off screen. It was the shelf edge we were heading towards. So you can look, great view now looking down in that false colour image, as we call it, the orange to green colour ramp we use to highlight the different water depths down the canyon. That's a great view back down the canyon. So you can imagine sliding down there on a slippery dip kind of fashion into the depths. Okay, that's a nice view as well. Perspective view. Showing what's to come later on this dive. As we're definitely going to go steeper. We'll see how that looks. Whether we have a repeat of yesterday's sheer rock face remains to be seen. Quite possible.
Fish up there, less pressure.
Just had a question online there from the YouTube chat as to whether there's any faults on this shelf. So, short answer to that is no. If you're thinking of before when we had the bathymetry view up and that nice along shelf view of the edge of the reef, you might be wondering whether that's a fault scarp, but no, highly unlikely that that's the case. What you're looking at there, that's escarpment, which is 200 metres high, is really the history of vertical build-up, vertical accretion of the reef, the Great Barrier Reef, over geological time. Multiple cycles of reef accretion over multiple cycles of sea level rise and fall. If you had the opportunity to have been online yesterday on our dive, we had Jodie Webster and Rob Beeman speaking to that history. So you could go back to yesterday's YouTube video and have a bit of a listen to that. But for now, that's not a false scarp as such. Having said that, we do know that there are deep-seated faults which sit much, much deeper below the seabed, which relate to the long-term, the million-year, the tens of millions of years history of this part of the Australian continent. Going right back to a time when Australia was part of Gondwana, supercontinent, and then started to break up, separating from all the other large continents of that time, India, Antarctica, etc. And here in northeast Queensland, that breakup included separation from a block which now sits offshore, forming the Queensland Plateau, subsidence of that block. And all that long tens of millions of years history definitely would have produced large-scale geological faults, but they're very much deeper. We don't see the evidence as such to the seabed in this area.
you want a reference for asking this, that camera right there. Okay. That's on both sides.
Okay. Yeah, I get you. Yeah, it's just,
So you want to focus for something else for your own.
yeah, beauty. Thanks.
Where do I type? 17.
Yeah, just noticing a bit of a chat online there on the YouTube side of things about the altitude we're maintaining, steady course, steady height. Yes, we are following an altimeter which is equipped to the SuBastian ROV.
That's my dad.
Yeah, and we need to maintain that elevation because that then gives us seabed photos of a constant scale, and we can do our measurements using the two fine laser pointers, which are green in colour, which are tracking along on the seabed right now. I'm not sure how visible they might be on your screens, folks, but they're midfield. Two tiny dots, green dots, which move along. They're 10 centimetres apart on the seabed, and that allows us to measure things, of course, give us scale. That's important. We maintain that altitude and constant speed because we've got a constant flow of photos being taken with overlap. So it's important we maintain that constant height and speed to do those measurements. And that in turn gives us a very robust data set, statistically robust data set to start to build our models of the way we think the biota in particular are behaving across the seabed as we've mapped it. So okay, you're getting these spiny urchins? Pencil urchins, thank you, Jeremy. There's a little colony coming in. Bit of a benthos party, definitely. Comment online.
You said you're maintaining a very steady altitude.
Oh yeah.
Is that automated?
Yeah, another comment online about the way we use these photos. We do build up a mosaic, a continuous mosaic of photos, which we stitch together in software. Pretty clever, easy to do these days, almost instantaneously.
Yeah, whenever we're on relatively flat.
And if you're really good at it, you can produce a 3D mosaic to really bring it into three-dimensional life.
Does the ROV carry two physical sensors such as sub-1 profilers or magnetometers?
We can, but we don't put it on right now. We'll put whatever the science requires.
You do sometimes.
Yeah.
Yeah, question online on the ROV's capacity to do other geophysical sensor measurements. Yes, I am advised. There's a sub-bottom profile that could be deployed, but we haven't had the capacity to do that on this voyage. It's a case of one or the other in many times. So
Certainly the ship has sub-bottom profiler as a hull-mounted system. 12 kilohertz for those online who are technically minded. But to be honest, that they become dedicated geophysical surveys when you start introducing that sort of instrumentation. Quite a different sampling design and approach to the way we work. Something I'd love to do, but it's all a case of can't do everything.
We should
Predict next, they'll say, yeah, this is pretty cool, but airborne is cooler. I'm calling that.
But at least your dad is interested in what's going on. My mom just makes comments about, yeah, you didn't do a very good job picking that one up.
Oh, in your imagination.
On the transect.
Yeah. How long in that transect, guys?
I'll have another 20 meters, 30 meters.
Seeing in your face.
We've already passed.
20 metre warning on the ROV Alex transect, 20 metre to go.
20 meter.
20 metres, so we're virtually there. Essentially everything you can see now pretty much will be what we have for the end of our transect. Then we will adjust to a new mode.
Same two. Did the same two fishermen follow us for like a half hour?
Pencil urchins. We're not seeing the lines anymore, are we?
Yeah, thanks, Jeremy. Good call. We've lost that those wavy, long, continuous lines on the seabed. They're still bite-debated, but yeah, not as intensely. There's hints of those lines. So what would we say about that? Our water depths are 320 metres. Yeah, interesting. They haven't gone away, but they're not as sharply defined.
They're not doing it for fun. They're doing it.
Still got a fair few little mounds, conical mounds of individual burrows. Fish is having fun. That's the same pair of fish that have followed us for quite a few 100 metres. They like the light.
Give it a little bit.
Scott, when you're scuba diving on the Great Barrier Reef at night time, it's quite common for red bass, which is quite a large predatory fish, to use the divers' lights to hunt smaller fish. So I'm thinking these guys are following us, just using the light, looking for anything to eat.
So they're clever. Yeah, that's neat.
Yeah. What's exciting is when you are doing a night dive and then you all of a sudden see a shark behind you.
That's exciting.
Also using the light.
Yeah. But you're the prey. You're not just using your light to find something else.
That's right.
See.
Yeah, it's quite excavated.
The only shark you need to worry about is the one you didn't see.
That's right.
I can see that. I can see that.
Yeah, those are good.
So this is not, and there's a little star maybe. Or, so this is a hermit crab pencil urchin.
Okay, coming pretty much to the end of our 500 metre. What's something lifting off the seabed in front of us? It's an octopus. Yeah.
All right.
No.
The transect, right?
Looks like an octopus. It's an octopus.
We went up to
Octopus on the...
Yeah, octopus on the... Yeah, we got him.
We just end the transect?
Yep.
Or Okay.
Yeah,
I'll stop. Transect's over. So we could look.
transect done. We're going to spend some time saying hello to this octopus if he stays.
And get distracted.
Like the ROV guys control.
And it's gone down to
The 16th going from here to Sydney.
you're doing maintenance course.
Yeah. Sorry.
He's kind of shrunk down.
Yeah.
He's got his eyes.
Yeah, it seems to be hiding.
Trying to hide, is he? Okay.
Hide from his metallic predator. But we could zoom in.
Hold on, I can get closer.
Here's a jellyfish.
Oh, is it?
Can't see it with the pages.
Okay.
Now, just say,
Oh, there he is.
get here in front of us.
This is kind of got it myself.
There we go. Come down. There we go. Yeah, it's not big. It's a tiny little.
You could zoom in if you want.
Yeah, okay.
I might fire bird back.
It's lifting off. I haven't got. Oops. Come back.
All the way.
Okay.
Alright. No, hold on. So we had just sat down. Now it's going to be
Oh, down a hole. Okay. He doesn't like us. Whoop. There he goes. Okay. Just keeping an eye on. Still. Interesting behaviour.
It's still there.
Oh, no. Oh, gee, no. Cancel that. Yeah, got back. So that would be...
He's.
What was oops. Where do I... How do I end them?
By the way. Down is up.
The down is up, yeah. Come on, bro.
Here, we do it.
Yeah, you do it, you do it.
That's the miles front.
I never played video games.
Oh, there it is. Oh, go.
One or the other. His adrenaline levels are probably pumping if he has adrenaline.
Oh, it's tucked away.
Leave me alone. Alien versus UFO.
Now let's get a lot closer.
Okay.
Alright. Yep.
Okay, let's
explore.
Let's explore. So we'll leave him alone or her alone and push up slope. Got a ways to go.
What's the range you want?
Okay.
7.5 meters.
Oh, it's pretty close. Max range.
317.
15, 30 meters.
Yeah.
Very cool.
Yeah. See if we get a course. Well, it's pretty coarse already, isn't it? Okay.
Follow the same line up.
Yeah. Thank you. The green colour is the edge of the data, isn't it?
Yeah.
Okay.
Get the sense that it starts killing, I reckon.
Yeah. We'll take a push call around about now. Thanks, guys. Thank you. So we're just about to land the SuBastian onto the seabed and collect a push core of sediment. The reason we've chosen this location is we know we're not far below the steeper incline of the canyon head wall, and this will be a nice site to get a sample that tells us a lot more about the nature of the material, the sediment being washed in from the Great Barrier Reef lagoon, coming down the steep slope onto this location.
Front and forward. Yep.
So it'll go out to and then come back.
I didn't think you're back last time. So. Oh, man, yes.
For those watching this for the first time, I know I've got a lot of followers who've seen a few of these, but perhaps there's someone looking for the first time. What we're about to do is take this Perspex tube, it's about 25 centimetres in length, and just simply use the mechanical strength of the manipulator arm to push it directly into the seabed. With the hope that there's enough cohesion within the sediment itself that it stays in the tube. And we've had a pretty good hit rate in the last few days with these, and judging by the amount of clay being kicked up into the water column, this is certainly fine grained. Quite a tricky operation for the guys, the ROV pilots, and hovering the ROV, which is the size of a car, and fighting the currents, and then just steadying it so that you can then use the leverage on the arm to push down. The sediments are very soft, so it's not. If you were standing on them, you'd sink. But that went well. So now the moment of truth as we pull it out, give it a bit of a wiggle to try and break the contact at the base of the core, and then draw it out slowly, fingers crossed. And that's a good one. Yep. Just in time.
Can you make it? Forward. Down,
Okay.
down, 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 down,
That's-
down, 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 down. Half just a touch.
That's
Yeah.
okay, that'll do. So we'll call that a 50% recovery. We'll take a second one. And that's not uncharacteristic of what happens when you've got coarse grains like we do have here. Very coarse, couple of millimetres, maybe a centimetre across. That just they just reduce the cohesion of the sediment, so it just fell out like it did. So it'll take a second just to give us a bit more material to work with. Obviously it's a disturbed sample, so we can't read too much into the fine grain layering patterns in this case. So we treat it essentially as a surface sample, integrated surface sample that we take as a bulk. Yeah, that's great. That's a beauty.
I'm seeing halometer.
You got a halometer, Rob?
Yeah,
Yeah,
it's
good.
just up in the top right.
Yeah, I thought so.
Yeah, 320. It's getting all the way down.
Okay, that's a nice plug again. We'll just cross our fingers that it sticks in before we get it into the canister. And we're looking pretty good this time.
All right, inboard.
I won't speak too soon.
All right, inboard. It's going down. Up a little bit. Up. Down. Don't worry about it.
We'll take it. We'll take it.
Get in there real fast.
If you can manage.
Higher. Back toward you. All right, that's a little bit now. That's a little bit high. Bring it in. Up a little bit. Reefs. All right, when you don't, you got it. All right. What did you scroll? Well, hold on. I want to dump this, get another one. 13.
Yeah, well, yeah, it won't take long. We're just going to empty that. Try again, because we know we can do it.
We can do it.
We can do it. It's really challenging when you got to do it quickly. It's best not to rush these things.
All right, now let that clear out so that. Yeah, it's pretty hard. Follow the camera line. Down. All right.
We'll take that.
She's back.
Wow, we gave it a nudge.
Break the tension there. Thank you. So yeah, we've got a call. We'll work with that. There's no doubt. That's okay. That was a tricky one. Still not quite there. Great, okay, thanks. Thanks, guys. Okay, we'll lift off again and resume our exploratory transect up the canyon wall, head wall. Okay, so expecting a transition at some point as we come up to 300 metres approximately at the moment. Steepening of the slope. Still combination of mud and shell grit, halometer flakes from the reef lagoon on the seabed. Still very sparse epibenthos. Not a lot of hard substrate for our sessile organisms to attach to. But that may well change as we continue up. Little sea star sitting there. And
hard to say because we run a sample machine and existing stuff.
sea cucumber, I think. Sea cucumber, Rob? Yeah, it's definitely sea cucumber. No, we'll continue. No, thank you. Yeah. Yeah, it seems to be.
Well, we've got another 100 metres of slope into us. It's hot, so.
In shallow waters, yes, definitely. Out here, there's these big solitary polyps, hanging down. That sea cucumber, Scott, they're using, they basically stick the sediment, like halometer and pteropods all over their body. They're really interesting. You see another one, zoom in. Yeah. You should do a big zoom in. Okay, yeah.
see that one? Let's see.
Yeah, we'll zoom in, yeah. If we do.
Rich, we just updated your position.
Thank you, over there.
We haven't got far to go, Scott, before it starts going up.
I know. Yeah, we're tracking well with time so far.
Second one. Just
Well,
going
yeah,
to
I
make
think
sure
right now it's probably
climbing
good.
up into taking small steps.
Small steps, that's no problem then.
So, if that happens, what do we do? What would you do? Would you go to the mid canyon of this canyon?
Yes, I think so. Just to give us that full.
It's been really interesting. Like there's this very common zone, but then we've seen
Varieties.
we've seen wood.
Yeah.
You know, I think that's really quite telling.
Yeah, it is. Yeah, I agree. Yeah.
I'm looking at the map. It looks like due west.
These is probably bumped up, not real. If that's 100 meters between these wavelengths, it's going to be around the next wavelength. It's going to take enough to make it. So like 150 meters from now.
Sitting at that.
Just wonder if we might just stop and just come down and have a bit of a scan of this material, this white material. What can we identify? Is it shielding?
Yeah.
Or is it just Alameda?
One of the
Why
axioms. Yeah.
don't we just do that? Because very soon we're going to hit that wall.
Yeah.
It's all going to be very different.
Yeah. Okay. Happy to do that.
That's my idea. I just wondered if you wanted us a sample or is the push core enough?
Push core something. Yeah. Or do you want a more strategic? You can get a more strategic sample of that coarse grit.
Yeah. So I'm quite curious what it's all made of.
Yeah.
And we know it's Alameda, but is it shell grit? You know, there's a lot of
Right here.
open.
Yeah. No,
Four right.
yeah. Rather than a core, we'll take our scoop.
Okay.
Just-
You want that white map?
Yeah, that white moundy bit there. That'd be nice. Yeah. Thanks, Chris. So we're just going to land the ROV here because we are conscious we're about to hit the steeper wall. We just want to really deliberately sample some of this coarse grain material so that we can identify it as a source and a sink for our, or a final location for our reworked reef sediment. Rather than a core, we're going to take a scoop.
ROV range seems like I need to turn a little bit to port side to be able to maintain heading on position better.
Alright, just keep north of it then.
Roger that.
Just keep north of it. Alright, we're gonna do the scoop.
Rich, just to let you know, when you do your turn, we're going to keep you north of us.
Yeah, I'll not move, I'll just turn in position right where we are as best as possible.
Alright, better.
This looks like Alameda. These are more lumpy things. That's not Alameda.
We're on a pretty steep incline. Just to let you know.
She said she didn't really need a sample here, but I was curious about what.
Yeah.
Because we're seeing some fairly big lumps.
Yeah.
I imagine there are coral algae in it.
Let me,
There's lots of fine Alameda flakes. It looks like a real collection of calcareous class.
how's that?
Gravel.
Yeah. Watch out more, you can get it. Alright, do you want to use the bag or the scoop?
The scoop's got holes in it, isn't it?
Yeah. Do you use the bag?
The bag. I haven't seen the bag used. Let's try that.
Bag is pretty awesome.
Yeah. Okay. You like the bag? I like the bag then. Okay.
Do you just want the surface?
Just the surface, thanks. Yeah, Rob.
We good on the sip.
Okay.
So event seems like two to sound is better than water. I'll go try that.
Roger that, thank you.
Give the bag a bit of a clean first.
The Australian.
Oh, that opens out nicely.
On my out. You want the struggle, right?
Yes, please, Ben. Yes, that that white, quite fresh.
In the first 2 centimeters?
Yeah, 2 centimeters. If you can pull that off, that'd be superb. Yeah. I like it. We're down at 286 metres, collecting a surface scrape of sediment, which we can use to really look at the composition. This is what this is all about. The size is one parameter or descriptor we use in terms of understanding the energy, the currents that have transported these particles to this point, but also the composition. You know, what are we dealing with? Is it halometer flakes from the lagoon plus shell fragments? And if they are shell fragments, what species do we have?
Yeah, stick it in there, just go straight up. Gently.
Yeah,
Let it all just fall into the back.
yeah, good. We're working up slope. You can tell how steep this slope is here because they've got a little avalanche there going on. Okay. That's what I would call an ample sample. Yeah, that's great.
Stick it in 2 or 3 or 4. What do you prefer? Box 2?
Which number box do we put this in, folks?
Is the bag already sealed?
What we'll do is when he's putting it in, we'll spin it. It won't seal, but it'll twist
up.
and then try to like layer on top. So
Okay, yeah, that can go in too.
2? Alright.
Cheers. I like that. That's the first time I've seen that sampling kit on this voyage.
Catch some fish.
We can put a net on if you'd like for next time. Grab.
The ROV turning is hopefully done, and then we'll see if we get back to the original position where we're meant to be.
How does that think? I'm not going to pay out. Stand by a second. That's not. Yeah, he drifted off quite a bit, just doing that turn. Alright, that's letting us out.
Should be safe to go.
Twist it first, like spin it. Yeah, so like bring your arm over like that. Nope. Elbow out. And bring your forearm starboard. Okay, just look in, bring it in, back in the thing so that you're up and down, strip it down. And just spin it like that. Now before you do it, alright. Shoulder left and forearm.
Because it's
right.
lunchtime.
There you go. Keep going. Shoulder left. Elbow down. Alright. If you wanna bring it up a little bit just to get out of the box and then just spin, just to close the bag up.
Or you want to twist it?
Yep.
Oh, okay. Just fold it. Still. This is a bag. How, according to this manual,
I'll get the, there you go, that's it. Should I get it off the bag itself? That's fine. Alright, I'm gonna stay here for a second, just to let this ship get back over to where he was, because he jerked it pretty far.
Okay. Cheers.
It's 183 meters away.
Yeah.
I can look around this area though, see if there's anything interesting.
Yeah, sure. Okay. Quarter to 12. Yeah.
got all that mesophotic to go into. Yeah. Keep us busy for an hour, I reckon.
Yeah.
Anyway, we're certainly further up.
We're ahead of yesterday.
Was that okay? Yeah. But you know, it's interesting. We've got this course here, but it's going to be like it's going to transition really quick.
Yeah.
It's just going to go rubble, big pieces of the course rubble.
Yeah.
But being open, is it completely swept clear? Is there no mesophotic?
Yeah. Let's find
So
out.
there's just too much debris, maybe.
Yeah.
How's it going?
It's going good, Jared. We're just sort of weighing up how we're going with time with this one. We're going well. We're ahead of yesterday, that's for sure, time-wise. So certainly another 2 hours on this, eh, Rob? Easy, another 2 hours.
If it's seriously good, yeah, at least
That's right, we're standing.
At least another hour,
if it's all
maybe.
there, we'll just
might shoot
throw
some.
out and go.
Yeah. So we'll call it later.
Okay.
Yeah.
So approximately about still another 2 hours unless it gets amazing and then
Yeah.
I'll update the bridge about that.
Thank you.
Y'all doing all right?
Yeah. The sea whatever they're wrapping.
Just a little discussion amongst the team here, planning the rest of our day. It's all about time management on these voyages. Maximising your time, predicting what's going to happen.
Huh? Oh, Hancock. Did auto focus one? Did auto focus one? Yeah, he's moving. Just slowly. I can creep. I can go slowly. Just have to. Yeah, a little bit more.
So here's my question.
What is that?
This rocks.
Yeah. Any slack chat?
Slack chat today, team?
What?
Pretty quiet on it from the Slack chat.
Yeah, just that those eye-fin amberjacks from Kai, he's our one geologist. They're following the sub in hopes of snatching up code disturbed by the operation. You know, they're taking advantage of the light. Yeah, they're smart fish.
Yeah.
Oh, look at those brittle stars.
Oh, right. Little Star City. Yeah, that's a change. Brittle stars scattered in great number.
Lots of
Undoubtedly hundreds of them just in view.
really dense. Yeah. Yeah, lots of brittle stars. Really. Oh, this city is a show. Scott,
Yeah, mate.
from Kai, I think, that amberjack has a cookie cutter shark bite mark out of it.
Oh.
Yeah. It has a shark wound.
Yeah.
So you might want to just
Yeah.
mention that.
Yeah.
So because they're hanging around, it'll be back. There's another goon.
So it's out of its fin, was it?
I didn't quite. It's got a screen capture on it. Might be closer to the head. But look for it's a bite mark about that big.
Yeah.
So, 20 centimeters.
Okay.
I noticed it earlier, so let's see it again. Start focus on it. Alright, I think it's in. Also.
Yeah. It's
Okay.
on the body. So it's twisted around and it's quite a decent chunk out of it. Right? It's a big scoop mark out of
I got
the
it.
side of its body. So it's come around. And,
Yeah.
And this is the pectoral fin.
Yeah.
Pectoral? Pectoral fin. Yeah.
See how we go. Right, continuing up slope to 79 metres. Water temperature now 16.2 Celsius, so warmer again. And that brittle star colony seems to have passed us by, so that was a localised occurrence. Interesting. Just one spot of them.
Sometimes it just takes a little bit back and forth to get it happy again.
Sea pen coming in view. Is Jeremy around?
Go on it.
Sea pen. I'm not going to call that. It's a pretty common one, I think. Rob, what do you reckon? Don't know. We'll keep going. Yeah.
Here, coming up. Sorry.
Okay. More wood. Another bit of tree branch. Just went past the sea pen, but
I saw it.
Did you see it or did you sense it?
A little bit.
Did you want it?
I know.
there's another one.
Some fairly large bivalve grass.
We'll look at it.
Big, Big pieces of
Didn't hear, no. No.
There's a lot of halamine.
Big shell.
Oh, this is a
conch.
baler shell. This is a really big shell. Let's have a closer look at this.
We'll drop and have a zoom in to have a look at him.
The zoom.
Yeah, is this baler?
In the deep, we were seeing a lot of these bigger than this one even.
It's not a baler shell, it's something else.
That looks collectively big. It's like what I call a conch shell. That looks like what? Like a conch shell.
A conch. Yeah, that's it.
I think we need this in our life.
Like in Lord of the Flies.
Exactly. Rob, you want it?
Yes.
Yeah.
Thanks. All right.
Okay.
I think these ones, like the Pacific Islanders, they can blow sound out of them.
Yeah, I stood up.
A baler is more round, has a really distinct smooth opening. And it's sitting, it looks like it's sitting on the wood. Good size. I think it's got something inside it. Maybe oh, it's a shrimp. It's a shrimp inside of it. There's a lot of halamine.
Yeah, there isn't there? A lot of flakes.
Yeah. It's quite fresh looking. It's got all its markings.
Yeah.
When they're sun bleached or they lose their colouring pretty quick, so it's quite fresh.
I think, Rob, you were saying you were looking for a paperweight. Was that right?
It's a nice one. Good size, 10 centimetres. It'd be about that ready.
No, you're another one. Yeah. Maybe a little more.
Yeah, nice. So shrimp inside. Yeah. Maybe an anemone. Something on the bone on the inside.
Yeah, what if that's a sponge in there? That big.
Yeah, yeah. Oh, that's roughly 12. Oh, yeah. Have you had some food?
Yeah.
I'll swap out.
Go for it.
Yeah. Going to leave the hot seat for a bit and hand over to Jeremy while I grab some lunch. Cheers.
So, there's a lot.
What? Got some good pictures. Alright. There's the shrimp saying goodbye.
He's like, that's a great.
Thanks for letting me keep my house.
Yeah, exactly.
Alright, let's look for some corals.
That is so exciting.
So don't move that.
And I just forgot that I can put down trim on to losing it.
He's not limited by a wave place.
Oh wait, let's stop here. Is that a... Is that a... Oh, it's a sweat. Oh, no, I'm sorry. Sorry. I thought it was like a fish, like a frogfish. But no. We're at 270 meters, 16.3 degrees Celsius. We're going to be heading as high as we can go. which would be somewhere north of 100 meters.
Can't manage the colour palette. But I'm saying this, but the colour palette is the max and minimum of each surface. You can't go beyond that. You have to match it.
Oh wait, let me look at this coral here. The whip. It's probably a sea pen, but I'll zoom in.
Very funny.
Interesting. I'll pan right just so it's
I'm just trying to get it so it's straight.
yeah.
Angle is straight, so it's
I try not to pan left and right much. They disorient you. So it looks like a bamboo. Yeah, let's throw it in 3-4. Hold on, just a second to image.
zoom out, sit down closer.
Yeah, sure.
I'm attached to the string. But I pull the string pull me.
Yeah, I didn't get a great look. We're going to get a little closer, but very quickly I was able to glimpse what I thought were evidence of a bamboo coral. Certainly a soft coral.
What's up, buddy? How's your day going?
Just started.
Too early to tell. Form an opinion.
It's nice.
So that kind of suggests.
All right.
I actually get to go a little bit more.
Yeah. I'll just zoom in. That's all right. Just take a look. See what it is we got here. So it's on manual now?
It's on auto.
Oh,
And
manual? For sure. So it's on. Oh wait, yeah, okay. Yeah. All right, so it doesn't look like a bamboo. Could it be zoanthids? Could it be zoanthids on a sea pen? We'll zoom in a little closer. James says octocoral. All right. Okay. Why don't we grab it and take some images and see how big is it? We got lasers.
where are they?
Is it down more? I don't know.
Put your arm next to it. The reference. What? Are you doing a picture?
Yeah.
Okay, so I'd say it's about that long.
All right. I want to throw it in 3-4.
Right from the base.
Yeah, please. And we'll get to see what it's attached to.
Hopefully a nice rock. Look at the
So James, so you're saying they're pinnate. So that's usually what I'll look for if I'm trying to see whether the polyps are part of the coral colony or something else, is if there's a consistent pattern, along the stem, which would suggest that it's a part of the coral. If it's a bit more random, it might be something like zoanthids or something else that has taken over a colony. Is that right, James?
also.
Yep. All right, James, I'm slowly becoming a zoanthid expert. Thanks for that.
All right, right there. Right there.
So at this moment, it looks like the ROV SuBastian's writing a letter in the olden days with the feather pen.
Can you drop in? You right in.
Fortunate.
Yeah, the zoanthids can live on many different corals. It's very deceiving. You know, you think you're looking at the polyps of a coral when it's, you know, just the zoanthids who have taken over, and they do such a good job. So you were telling me that the tissue of the zoanthids connect to each other, making kind of one massive colony of these zoanthids, as opposed to the anemones that wouldn't necessarily fuse their tissue. And so James is listing off all the things that the zoanthids could live on. Octocorals, hydroids, echinoderms, black corals, nautilus shells, hermit crabs. Yeah, the worm tubes.
Good location.
Jeremy, are you doing 2 dives today?
I think it depends on how this dive goes. It's already lunchtime, so probably not.
It's sink or float?
My money's on sink.
Like, crazy now.
I said it as it started to sink.
Still there? Yeah, you grab some meat, bud.
All right, so we grabbed an octocoral. We'll continue our ascent.
We were looking at it down there. It's
Oh, yeah, we turned the lights off and the turtle shined on.
Crazy diamond.
Mm.
Shine on you, crazy diamond.
5 seconds ago.
Let's get you out of these shadows. Do that. Is it 5 seconds?
Okay, here we go. We have Gemma here to assist in the commentary.
Hi everyone.
Gemma's our onboard fish expert. When you have 0 fish experts on the boat, the one who can identify any fish becomes the fish expert.
That's about right.
So I'm by default the black coral expert. Looks like
Is that the sonar or no?
anemone.
There's something with the sonar. To the right.
It's pretty lonely out there.
Might be a rock. Might be a shipwreck.
What? Do you see that, Jeremy?
Oh, there's a coral here.
Just
Here, let's just.
Oh.
That was, yeah, good eye, good eye.
You just zoom out and get a reference of where it is.
Yeah.
Left. Alright.
Yonder. So, yeah, we'll see what it is. It's hard to tell when you're far away. It seems branched, but it could just be shadows from the ROV.
What is the adjustment?
Oh, it looks
I
branched.
think we got branches there. Very fine. Oh,
Pin 8.
yeah, okay.
But it's still interesting. If you'll remember yesterday, we collected, actually, at about this depth, we collected something that looks like a schizopathid, so a black coral that has these alternating pinnules. And I was saying it's really odd to find them so shallow. And this looks like something similar. So we'll just take a look. If we could move 90 degrees around to look at it head on.
Just me,
Yeah, all yours.
no. You want to look from our right side?
Yeah, this way. So we just want to see straight on the stem, and see the pinnules on the left and the right.
That core right there, that direction.
Yep. And so we're going to look to see if there's any interior branches between these two longer branches. We're slowly falling away, but we're heading back. There you go. Where was it? Here?
Yep. Yep, that's it. Just it.
Yeah. Over here.
Ah. You want to come at it from the side, then?
Yeah, so like sitting here, looking at it that way.
Where's that going?
Yeah, it's really small and fine branch, so it's hard to see.
You see Jeremy? Oh, right in front of us. Yeah, right in front of us.
Oh, yeah.
It's right there. Yeah, it's really small. Yeah, these things are interesting. So I bagged one yesterday, and it was really small. Still not really sure. I guess what it is. The depth is, but it's throwing me off. So, are we down?
Yep.
Okay, I'm just gonna take a close look.
Going into an auto turn.
Okay. So we'll just zoom in.
Yeah, okay. Seems very similar to the one we collected yesterday.
Yeah. All right.
So fine.
Yeah, let's try to zoom in. We could see the polyps. All right, I'm gonna. Is it on? It's on auto now.
Yes, sir. It is on auto.
So if I click the auto, it'll go to manual.
It should. And it's in man.
Disturbing disturbing little. Yeah, excellent. Thank you.
Here you go.
I'll
Close
just.
the back one.
So you could see. We're almost there. Why don't we pick it up by the rock, and we'll take a zoomed in look from that way. That'll be easier. Yeah, all yours.
Of course, the little crab's gonna get right in my way.
So the features we're looking at here are the distance between pinnules, so distance between these branches on one side of the stem, the length of the pinnules, so you can see on the bottom that they're short, they get longer, then they get shorter again. And it's a simple coral, there's no other There's no other branches or sub-branches or pinnules on the colony. And as it's being picked up, you could see that it has those 6 tentacles. But we'll take a nice view as the arm is brought to us.
I'll get it right and I'll like Jeremy.
Yeah, man.
There you go.
Thanks.
Very nice.
All right, let's just look close. It's really close. I'll try to.
Give me a talk in manual then.
Is it on manual now?
It's on auto. It's on auto. There you go.
I'll zoom out a little bit first. Okay. Cool.
Hold it there. I can bring it in. There.
All right, I'll just zoom in.
Pretty translucent,
Yeah, very thin branched. But this, we should be able to focus on this. Another way. There we go. All right. So, you could see little crustaceans, really small crustacean on it.
Yeah.
Beautiful. So now we can see the distance between the polyps as well. All right. Yeah, it's a really interesting colony. So we'll throw it in a bio box. What do you think?
I don't know what other tools are in what position, but
It'll fit into it for sure. Want to throw it in two?
$12 too.
Yeah.
And we got a rock sample too.
And we got a rock. So the
description on this journey.
schizopathic. So there's a question about the age of the colony. So black corals are slow growing, but having said that, it's more of a general description of the growth rate of the black corals. Of course, different species grow at different rates. The shallow ones are faster growing than the deeper ones. Generally, we just don't really know. So, yeah, it's hard to say. Could be young or could be old and small. And so we're coming up here to another coral.
A little bit bigger, this one.
More
Okay.
sea pen shaped. Yeah, looks like a sea pen. We're gonna skip that one. We've collected sea pens.
Starting to get a little more rubble up this slope now.
Yeah, I imagine we'll start seeing more corals. Yeah, let's do it.
Flying up.
It's going on the same possibly this tip here.
Another sea pen to our right. Oh no. So what black coral it looks like? Oh. Gorgonian? Yeah, soft coral. No.
Do you want me to do kind of like a up the hill?
Yeah, so
Cover
you
the
can,
same, kind of most of the area there?
sure.
Okay.
Sounds good to me.
I'll just kind of slide kind of left and right all the way up.
Right, that'd be awesome. Yeah, we'll look for some interesting corals. Just so I'm in sync. Right now, are we in
You
auto?
are in manual.
Okay, good. So I'm switching it to auto. Yeah.
Now you're in here.
Cool. So there is a question about dating these corals in the lab. So certain corals, like black corals, have rings, like essentially like trees do, but they're not super consistent. And you could imagine that if the rings were made due to seasonal changes, in the deep there aren't seasonal changes every year, right? The temperature is the same. So there are rings of growth, but it's really hard to determine what it actually means. And then there's ways to determine through chemical composition, I think, the age. But from the people who are working on it, so NIWA in New Zealand is working on these kinds of questions. And it's just not a very easy question to answer at this stage. We're still trying to figure that out.
This is interesting. It's like
Is it a sponge?
sponge, I think the wrong way up. I have a feeling that should be a vase.
It's an upside down sponge.
Vase shape, yeah.
Pretty neat.
And there's that's all right.
Can you imagine? Got our amber jokes back.
Oh man.
It's got a hole in it.
Cut a hole, is it? Yeah, let's let me see that again.
I'm sure we will.
I've been following us for a long time.
Yeah. So we're seeing more of these sea pens.
Yeah, I guess the ROV is just like a nice big fad for these fishes. They're just hanging around, cruising along with us.
A light source.
Maybe feeling nice and protected by a much bigger
That's right.
creature.
Yeah, they're like remors on our whale shark.
Yeah, just thinking about that sponge, you know, can you imagine being up a couple of 100 meters up and tumbling all the way down this slope?
Right. Yeah, well, it's happening, right? You're seeing all these shallow things down here.
Oh, okay. Yeah.
Cup coral there's you.
Oh yeah, some cup corals.
Yeah.
There's a tube worm.
Little fishes.
Can you identify these fish?
Looks like there's some kind of gobies just perching on the sand down there.
I'll take that. Goby.
There's a little shrimp.
Yeah.
Oh, these guys, these are
This
not
looks alright.
gobies.
We'll keep going.
Wow. Oh, you got an orange.
Nah, keep going.
See?
Nice thing.
So we're seeing lots of rubble.
Yeah, a lot more fragments of stuff we're beginning to see.
So can we assume that this is the stuff from the shallow that's died and fallen?
Yeah.
And can we also assume that we're going to see a lot of living stuff in the shallows?
So we're making our way up this canyon and ridge system, and I guess unlike yesterday where the top of this canyon was blocked on the shelf by an emergent reef, when we get to the top of this one, it's actually open to the shelf, so it seems to make for, you know, potentially a lot of stuff washing through that channel, coming down off the shelf.
Let's kick it up.
So yeah, whether that stuff is right above or maybe
on
coming
this-
from
the canyons.
other parts of the reef.
So Jeremy, is
Hey
there
Luke, while you're changing that heading, I'm gonna actually drop
quite a shot? Yeah.
Are we sampling right now? Or?
Not this second.
Okay.
Do you want to grab some sediment?
No, not as such. I just think we might kick along a bit along the transect and start climbing. If there's.
Yeah, sure.
Yeah.
We're seeing the odd coral here and there, but certainly we could start straightening our course upwards.
Interesting, Marty.
Yeah, just talk to comments about it.
Wow, it's
very,
such a defined line, eh? It's like it's almost like
it's like a runway.
like a tractor has ploughed a line along the field of this
Yeah.
shaly stuff. It's pretty amazing.
Waypoint 11's our last one, isn't it? Is that right?
Yeah.
Yeah, okay.
The second dot is 1300.
Yeah. Yeah.
Ooh.
Yeah.
So
Here on this
coral.
one?
Yeah.
Yeah, maybe
Fleshy.
maybe it needs to be 130.
131 deck.
I'm thinking it all depends what we see in the next little while.
Yeah.
Right?
We
If
could.
it's really good, we'll just take the rest of the day on this
Yeah.
one.
No, we could zoom. We could zoom as fast as we can up this wall, and if anything stands out,
Yeah.
we could change our plans. But otherwise, we could just
Okay, let's
work our way up. I mean, I assume in the shallows we'll find something,
yeah.
but we could skip this section.
Okay, let's
Flying along now.
major feature.
Yeah.
Ah, so that's a cool view. You can kind of see either side of this ridge.
Blocks.
Okay.
Oh wait, is this branching coral? I mean, it's probably dead and falling branching
Yeah,
coral.
looks like some.
Oh wait, can we slow down here for a second? So, if it's fallen down,
Is it a
it's.
sponge?
Oh, okay. Yeah. Who said coral? All right, let's keep going.
Branching.
Rubble.
Oh yeah.
Oh, rocks, slabs.
Landslide.
Okay.
I think that whole.
So it's shiny.
Looks like a shell. Shell off to the right.
The rubble zone.
What's this on the right? What's this on the right? Oh, it's a baler shell. This is an empty baler
This is a
shell.
baler. All right.
Yeah. So Pacific Islanders use this shell for bailing out water from their their canoes.
Keep moving up.
It's old.
I reckon someone's dropped that from their canoe.
Yeah.
Yeah. I've
Someone-
seen live ones. The animal was huge. You know, the foot on them can be, you know, like a good 30 centimeters across.
Amazing.
Okay, so we're seeing a lot of rubble. This is, you know, like a big scree slope, but then behind it, this looks more framework like. This is what Jodie Webster was talking about yesterday, where in situ coral, but you know, highly eroded. There's lots of cavities in it, but it's a more solid shape as opposed to this layered scree slope. So we're going to see more of this as we go up the slope. There's lots of animals living in the cavities, so let's just keep an eye on that. Okay, we're right on the wall now, aren't we? Still a lot of sediment draping everything. Big boulders on the right. We've got some fish.
Just on the screen gives you a pretty good.
Fish to start under that little rock. Some hermit crabs.
Yeah, is that another branch? Another like tree branch? We've seen quite a few on this particular transect.
Yeah, you'd imagine they'd be up here as well. Yeah. What's
Oh, is it? What's that on top of the rock here?
We'll slow down for a second. Past it.
Okay, lots of sediment draping the rock. Oh yeah, look, there's a big, like a big screw slope coming
All the
up.
way out.
Yeah, so we've seen lots of these shoots. We call them region spur and grooves, and this is a common feature of the front of coral reefs. Yesterday we saw these great big bluff cliffs, and they were separated by grooves that literally cut directly into the rock, and they become chutes for all of the sediment which rains down from above. But in this case we were looking at spur and grooves that were 100 metres high. That's quite something to see. Yeah, you can almost make it out in the 3D view of the inset. So we're heading up, we're at the very base of this wall, and you can see all these sort of stripes, these vertical stripes along the face. Now these, we believe these are real, and they're probably the spur and grooves that we were looking at yesterday, and we hope to see here. So what we're saying is this is not a big, long, flat wall, a rock face, but it's actually interspersed with lots of these spur and groove morphology. Yeah, so if you go onto Google Earth or get some really high-res satellite imagery, like the Allen Coral Atlas, which was developed by
Right now, yeah. You want us to try and break some of that off? Yeah.
that was developed to look at
These little
high resolution,
these fingers sticking up. Let's try and break one of these off. Sure.
high resolution satellite imagery over coral reefs. You can see these spur and grooves often at the front, the windward sides of reefs, and they're really distinct. Yeah, all right.
Yeah, that's good stuff.
Oh, there's amberjack.
It's not amberjack backs. They've been with us for a while, and it's the same guys.
So Robin Beeman from James Cook University in the audio seat here, and we're just starting to climb up this steep wall, seeing all kinds of things. Probably big plate outcropper just sitting here on the left-hand side. But we've decided we're going to see if we can break off a piece of rock. We're now at a depth of 210 metres water depth at the very base of this steep cliff. So as we learned yesterday, the difficulty of trying to get a sample on these steep faces, you have to somehow park the ROV on the seafloor so that you've got a so you can get a good purchase position. And we're just giving some instructions to the ROV pilots. Brandon's driving the manipulator claw. Because we don't want just any old rock. We actually want a bit of solid rock that's attached to the seafloor, not just rolled down from above. Otherwise it kind of confuses the depth-age comparison. So we'll just see how loose this one is.
Pretty easy.
In particular, we want a nice fresh surface, something that's broken off.
Yeah, there's another one right to the left there.
Can you reach in there?
That'd be easy for you to get in and up. I can just bump up to it.
Okay, we're going to try for another sample. If you look up in the top left there, there's a little bit of a split in the rock. Yeah, it's quite pitted and eroded here, but we're looking for something fairly solid that we can snap off. And that way we can be sure that it's actually coral that's in situ, perhaps grown in situ rather than something that's just tumbled down from above.
Right where those lasers are?
Yeah.
Okay.
Yeah, Jason from Michigan was just pointing out the harp comb jelly that we saw yesterday. That's a benthic ctenophore, really large, sitting on the sediment. And yeah, we agree with you. That's quite a sight. It was a strange looking animal anyway. We've got Jeremy and our other biologists here. We're all looking for benthic fauna attached to the seafloor on this drive up this rock wall, because we have the mesophotic zone above us. Big question we have is just how dense that might be. We are doing a really interesting comparison here between a canyon that's directly in front of a reef, a big barrier reef, and that was the one we did yesterday. And I think you'll agree that it was spectacular. Lots and lots of soft corals and things like that. Really quite beautiful. This one is in front of a big reef passage. It's about 45 to 50 metres deep, and it's the big gap that lies between Ribbon Reef 4 and Ribbon Reef 5. So it's completely connected to the shelf. We've already seen evidence that there's different kinds of debris in here. We've seen quite a lot of pieces of wood and timber, logs in here, and even some rubbish, unfortunately. But we really have no idea what kind of marine life we're going to find higher up on the mesophotic zone. It could be completely swept clear, it could be barren, or it could be a biologist's delight. We'll find out soon. We've got Gemma sitting next to me, and she's keeping a sharp eye out for anything worth keeping. This is our second coral fragment, limestone fragment. It would be fossil coral. We have our very important collaborator, Jody Webster from the University of Sydney. He will take these samples. They will be cut in half. We get a nice clean face, and then we can start doing all kinds of work on these rocks. For a start, you can actually start looking at the composition of them, like what kind of coral or algae might be making up this fossil limestone, even to the point they can get down to genus, sometimes even species, just from looking at the fossil, the sclerites, the internal skeleton. So I think that's pretty amazing that you can see this fossil coral and work that out. The other thing they can do is a lot of geochemical work, and that just so many different things you can do here with that. You can look at the strontium-calcium ratios. You can work out proxies for sea temperature when these things were living, and of course when they die, that signature is locked up in the coral skeleton. This is not my field of expertise, but Jody tells me you can learn an awful lot about this past environments, the past oceanic environments, just from this very, very fine scale geochemistry work. And of course the age dating too, which is important, and we can relate depth to age then and get a sense of the growth, you know. And at this 200 metre mark, we're really talking about the very, very base of the Great Barrier Reef.
Okay, target 130. 130, yeah, okay. Target 130. Let's track up to 130.
Go ahead, Luke.
No, yeah, we're having a lot of troubles making any sort of lateral way to starboard, so I'm going to have to change the heading again, probably 15 to 20 degrees to starboard. But I'm not able to come to that position there at the moment.
That's fine.
Just tell them to stay there. I'll come in behind it. We'll just.
How about just there?
Yeah.
You good with that?
Yeah, let's go for that.
Okay, Luke, you make it happen, Cap.
Right, yeah, I'm just funneling through these two reefs at the moment.
Is this
so.
the ebb tide coming out?
Yeah, that's fine.
It is.
Just keep us updated. If we've got to alter our course, we definitely can. I don't think we need to sail in this sort of track line. We're just trying to make it to the top.
Okay,
so
that's good.
We'll just keep you updated and let us know.
Okay, cheers.
I'll just slide in behind him.
Yeah. Is that okay with you all if we just slide in behind him and just keep coming up? Instead of following this line, where there's some great importance about this line.
No,
this is good.
Just get to the top.
Just get to the top. Get to.
Yes, you might have heard some discussion going on there. It's always a delicate dance between the ROV, which is 4 tons of down on the seafloor, and the ship. And we were just discussing with the bridge team here just how to manoeuvre the ROV to make it a little bit easier for the ship. So at the moment the ship is pointing east away from the Great Barrier Reef. But the ROV itself, we're climbing up this wall, which is a passage between two shallow coral reefs. It's also ebb tide, which means the water is moving outwards, so there's that to contend with. But thankfully we have really great weather at the moment. It's absolutely flat out there.
So next target will be 130 metres.
Okay.
Unless we see biology on the way, but.
Wow, that is a big pile of rubble. You can just imagine that has collapsed. You can see all the different plates. Not saying that they're individual coral colonies, they look a bit big for that, but just look how that is just torn down the slope. They're all just stacked on each other. Yeah, big scree slope. That's really interesting. We saw some boulders back before. They really did look like boulders. You can see some of these just sort of perched on top of each other. Very, very bouldery to me. Yeah, really quite amazing. So the ROV is actually tracking, I guess, north along the face of this wall, which is in front of the passage. We'll call it Ribbon Reef 4 and 5 passage, for want of a better word. So we have shallow coral reefs both north and south of us, and yesterday's dive was up Ribbon Reef 5, which is the northern one of those two reefs. It still looks like a very, very big scree slope. Lots of very coarse material. Not seeing a lot of benthic life, seafloor life, and it could be that it's just there's so much material raining down from above that it's scouring it clear.
Looking at how far we still have to go to get to this top. If you want to just hold position there.
Okay.
And we'll just sneak up behind you and just continue straight up. If that helps you out with this current situation.
Okay, yeah, I might still have to bring the heading a little bit to starboard, but we'll try to hold here.
That's fine. That works for us. So we'll just continue moving up once we get behind you, and if you have any issues, just let us know.
Okay, cheers.
That's what I want to.
So we've brought the ROV a little bit closer to the ship, to the Falkor, and we're going to start moving up a little bit more. Now we're at 200 metres.
Someone cruising just in the...
Yeah, it looks like we've got a couple of large fish in the off in the distance. I'm not sure if they were those amberjacks we saw earlier in the dive, but we are still going over a really big scree slope. Lots and lots of debris, layer upon layer of clasts, we call them. and it's hard to even see perhaps in situ coral. In fact, we might be on some here, but most of what we're looking at just seems to be boulder pieces and great stacks of debris. And of course a lot of sediment too, and we know that does suppress marine life. There's a few sea whips, but they're actually attached underneath the overhang. It's got to make sense. Here we have a look at this coral. It's right on the... I'll just swing left here. This is in fact a big colony, right? You can actually see perhaps I don't know...
This one here?
Lobophilio, potentially?
Yeah, thanks. If we can just have a look at just close in here. This is a very large single colony. The front of it's actually broken away, and this would... I'm not sure what kind of genus we're talking about, but the coral, the actual polyps would be very large on this one. You can see how they radiate out from the central part of it. Of course, when you scuba dive on the Great Barrier Reef, you can see these kinds of colonies quite regularly. But that is definitely a big coral colony, and here we are. It's at 200 metres, and it's rolled down the slope. That's unequivocal. That is a big coral colony.
You want to zoom in on it here? Welcome to stay on.
Thanks. We can keep moving up. Yeah, it was just to have a bit of a look at the internal. Yeah, thank you. I wonder what that is. Yeah, this black piece here might be just a bit of veg. Oh, is that a bottle? Bottle, or it might even be just a piece of a branch.
Trying to see what's going to be best with these currents because they're coming from a stern. So we're going to try an alteration to port.
Okay.
Large alteration, probably 25-30 degrees.
Okay.
See how the ship reacts there. So just to let you know, yeah, we'll be coming the other way now.
That sounds good. What kind of currents are you seeing?
Over not a current, setting kind of northeast.
Okay.
So it's basically coming right from behind us, which makes it challenging to keep position. But yeah, it's quite strong.
It's a tidal
I think we're
jet.
on the ebb of the tide, and it's just coming through those through 4 and 5, and just kind of funneling through those reefs.
This is good.
Good fun recovery.
Yes. Yeah. Exactly. It'll be basically the vehicle will be coming into the vessel, so we'll have to be careful.
Yeah.
Yeah.
All right, bud.
All right. Cheers.
So you may have just picked up a conversation going on between the control room pilots and the bridge staff. This channel that whose rock wall we're in front of is an inter-reef passage, and they are. We well understand these. This is where we have these tidal jet pairs. There are every ebb tide. We have a tidal stream moving out through the passageway, and where the vessel is sitting, you get this kind of twisting effect. You get a pair of vortices. Now, it's only a shallow water vortice, but it's affecting where the ship is. And on the flood tide, as the water moves back onto the Great Barrier Reef, you get the opposite effect. So they're called tidal jet pairs. And we actually know a fair bit about this because we have Marty McNeil with us, and these tidal jet pairs actually do have the effect of bringing upwelled cooler water onto the Great Barrier Reef shelf and nurture these Halimeda meadows that sit just behind the Ribbon Reefs where we are now. So it's a really interesting oceanographic effect, but it's driven by the tides. And here we have diurnal tides, so 2 high tides and 2 low tides per day. And we're just at the end of an ebb tide, and the ship is saying that they're getting a knot of current, one knot of current pushing against the ship. So. But we expect that will probably ease off in the next half hour or so. And I think we've got some antheas coming in. Right, so we've got our fish army watching. I think that might have been the first sighting of the Antheas at this depth. So we're at about 186 metres.
Potentially some CZonids, so we're talking about currents here, Rob.
Yeah.
These CZOs, they sort of love hanging out in those strong current areas, so the current features are going to be really interesting to see how that affects our fish life.
Yeah. So talking about tide range, it's not huge here, but we're talking around about 2 to 2.5 metres today. During the spring tides, it can get up to about 3 metres. So we're not talking really big tidal range, but because we have these almost like these two reefs, or a series of reefs. These are the Ribbon Reefs. You know, each one is a complete barrier to any water movement backwards and forwards across the reef. And so the only place for that water to move is through these inter-reef passages. But it just has these very interesting effect on the on that surface water, where it actually coils. You end up with these vortices. It's almost like, if you're looking from space, it's almost like looking at, like the feathers. You know, what you've got a twisting one to the north, and you've got a twisting one to the south. And we're still seeing a lot of debris here. But okay, this is more like sort of in situ coral, and it's more layered, and some terraces, because this is exactly what Jodie Webster was really looking for yesterday with these low stand sea level indicators. That would be a little bit further up the water column. We'd expect to see more of that around probably around 130 metres depth. So we're now at about 170 metres. But we were talking before about these ridge and spur grooves, and this is classic ridge and spur groove, but I've never seen one from, I'm used to seeing them in imagery, like satellite imagery, and talking about you can actually scuba dive and snorkel over them, but it's one thing else to see them from 170 metres and up. Just the sheer scale of these spur and grooves is pretty amazing. But you can see how the grooves have just become the rubbish chutes for all this debris. It's all sort of funneled into these big gaps. Oh, and lovely overhangs here, guys.
Nice.
Yeah, really interesting. Now, yesterday we got lots and lots of soft corals underneath these overhangs. I'm not seeing a lot of that here, though. Maybe it's still a bit deep for them.
Rob, put a picture of the sonar, forward-looking sonar, so you can see some of the...
Yeah, okay. Yeah, thanks, Viet. We've just put the forward-looking sonar on. This is a safety sonar. It's just scanning... and it gives everyone a bit of a sense of where the hard stuff is, because we want to keep off that. And you can see how as it sweeps backwards and forwards, those bluffs, the ridges are showing quite clearly in the scanning sonar, and you can see the spurs as well, right? Those are those indents. So imagine if you're standing looking down, that scanning sonar is just sweeping backwards and forwards in front of us, and if we had the lights off, we would still be able to see this picture, this sonar picture, because it's using sound rather than light to generate it. Now, one thing that's becoming obviously, you see this red coloration on the exposed limestone. This is red coralline algae. It's a crustose algae. It's got red pigment in it, and it indicates the lower depth limit of light. So these photosynthesize.
Okay.
And then once I get there, I might come even a little more forward, but I'll let you know.
Yeah, that's fine. If you come forward, just keep in mind, like, around this from here, that waypoint to that waypoint is fine with us. So from waypoint 9 to Falkor waypoint,
okay,
you can play in that area. I probably won't move you after that. So whatever you feel good in,
Okay.
we'll keep you there.
Ideally, so right now, where would you like me to go? I'll try to stick relatively close.
What's easiest for you? Just straight astern? Or kind of off to the west there?
You're gonna make a
Yeah.
port heading change?
Yeah, anywhere kind of directly astern or even off the starboard quarter.
First mesophotic.
You just move kind of back towards us in whatever direction's best, and we'll line up behind you.
Okay, great.
Cheers.
Cheers.
I'll just throw that there.
Soft core. Okay. Yeah. Get these guys. Gents, can you just go to the right, and we'll just have a look at this. This one over here. Can we just have a look at that, please? Oh, yeah.
What I'll do, we're just gonna, I'm gonna land out here, I'll let you look around, we're gonna let him kind of catch up with us.
Okay.
Get tossed around, so.
Finally, some life on the rocks.
Yeah, so there's still we're still a fair bit of discussion between the pilots and the bridge staff just to get ship positioned and everything. So because we're in this tidal jet stream here, but this is really our first real glimpse of benthic marine life up this wall. It was fairly barren until we got to here, and it's really kicked in at about 160 metres. So I've got Jeremy Horowitz next to me, and we're just going to try and set the ROV down and have a bit of a look around.
There's a good view. We can look underneath this cave. There's this overhang.
Yeah, clearly the overhangs are important because there is so much sediment coming down.
Once he gets set up, it's actually not bad. It's actually blowing us in a good direction where it's throwing our umbilical off.
Yeah, it seems to be a real feature in the deep ocean. Wherever you have these overhangs, this is a real favoured spot for marine life to attach to. Doesn't matter that it's upside down. It's just they're just waiting for food to come past.
We're on the ebb right now. So when it switches back to the flood, then it's going to flow the opposite direction. That direction for us is ideal for recovering. Right now, if it's going this way, it's good for at depth because it's more umbilical back.
Yeah.
As soon as we go to recover, we have to go back and we're getting caught in. It's going to want to go to the boat. But if we wait after, then the tide flips. The tide goes against the boat, and if we go against it, it keeps us off the boat, which is that's the best idea. If the tides are small, like 0.5 or whatever, it's not a big deal. You can do it either way. But when you start seeing this right on the chart here, then it relates.
Okay. Let's just work out time tonight.
Yeah, we're just having a discussion about the effect of tides here. It's also going to affect us when we recover the ROV. We've got an outgoing tide. It should turn in a little while, and of course then the tide's going to be flooding back onto the shelf, and so that's going to influence the recovery operation. So everyone's trying to think ahead here how it's affecting the ship and the ROV.
Like you're saying, is if we stayed at this site, what's the latest we did it, or what's the
Well,
latest? Yeah, you'd have to talk to Jared because there's also an issue with the deck crew.
Yeah.
For recovery, but yeah.
I'll go with 6:30 for now.
Kicking around there.
So we're just having a look around here now, and Jeremy's looking for anything particular that we might sample.
I don't think anything just here.
Okay. And we're also waiting for the Falkor to reposition above us at about 160 metres above where the ROV is at the moment.
Feel free to move around. Yeah. We're just going to let him slide in behind us here in a second. And then once he's happy, we'll continue on. We just want to be in a good position once we crest over.
Yeah.
Currents and low shallow.
Yeah, so it's always a bit of a juggle between the ROV and the ship, and of course the currents are moving. You've got currents strengths at different levels. So at the moment the ship is being pushed around by an outgoing tidal current through this passageway. It looks quite nice down here though. So we're just having a bit of a look around, and as soon as the ship's repositioned, we'll then start moving up the wall again.
It's like one of those crystal gorgons we collected yesterday.
There's a lamp,
So-called crystal gorgon.
Yeah, heading towards the house of the ship.
How bad is it?
I think you're still okay, but
golden coral.
you gotta either move the ship forward or you guys move the other way.
Copy that. I'll let the bridge know. Thanks.
Crystal gorgon. The black coral behind it.
We're going to come to an all stop there if you can.
All stop.
All right.
Yeah, so we've got a black coral, that's that sea whip-like shape, and there's that golden coral, Crossagorgia. It's got something white, some white colony attached to the bottom of it. They have this classic sort of long feathery look, and they do have that golden colour about them. We have found them much deeper. I've seen them down around 700 metres out on Osprey Reef, out in the Coral Sea. Osprey Reef is one of our destinations in a few days' time. So we'll get another chance to see some deep cold water corals down there too.
Hey, don't put a lot of slack on it.
We're picking up, Ben.
Yeah.
How's it looking?
It's still inboard.
Okay. Yeah, just pick it up. How bad is inboard? Is it?
It's not comfortable for me.
Okay.
Yeah.
Can you jump in? Look, you see I'll sit here. If I see rock worthy tools.
Yeah.
Can you come like 20 meters?
Yep, coming ahead 20 meters.
Ben, is the umbilical also leading to vessel port?
What time are we?
Yes, a little bit. But it's more inboard. It's getting better now.
Okay, I'm gonna just push ahead here.
Yeah, it was like one of the two. It was only 15 minutes getting it out.
Okay, so it's getting a little bit better.
So we saw one section of rock that had corals.
Look at this cave. Look in here.
Hey guys,
Okay, see that? It's a lot better.
in this cave opening.
How's that looking, Ben?
Of course.
Yeah, it's a lot better.
Okay.
So, Rob, we'll just can the second dive. Just get our numbers on the times.
Just 22 seconds.
Yeah,
Slow down, take our time, because this is pretty cool.
Then we don't necessarily have to go there.
So you can go along a bit, yeah.
Yeah, let's meander.
Let's have a look around.
Yeah, well,
And that's a good thing, because the tide is starting to accelerate the higher we go. So,
okay.
Okay, that's good to know.
Yeah, Ben, just keep a close eye on it for us if you don't mind.
Roger, that's I'm here.
Thank you.
It's pretty good
So we
now.
got a nice
Okay,
cave
sounds
here.
good.
You imagine a nice hiding spot for fish, and I assume inside lots of corals. Wow. You could see some soft corals hanging on the entrance. Looks like a nudibranch.
Nudibranch,
I'll
yeah.
get you in as
Yeah.
close as I can, just so I can see what's
Yeah.
on the other side.
Yeah.
Perfect.
Okay. Yeah, it looks like a nudie. Big,
Upside down, yeah, it's attached.
hard.
Yeah, that's not the better. Okay, go for it.
Wow, that's cool. Wow. Whoa, go in that cave.
Yeah.
About as close as I can get, I think.
Oh, that's fine.
Yeah, definitely a nudie.
That's a nudie you'd see on the surface. Here, let's. Don't want to fight the. Oh, we're close. So we
How
gotta.
deep is that?
I can relax on the cool ones. I don't know,
Sandy seafloor, flat.
It's big.
Just, yeah, really big. Really big. And light's coming down from above, so it's definitely open at the top.
Right. You don't see much on the floor inside.
You don't always, you know, when you go to those swim throughs, they're can be pretty
That's
big.
that's about 20 meters in. So it goes about 20 meters back.
About 20 meters back.
That very last spot on the scanning. It's about to disappear.
Oh, yeah, right.
Disappear now. That's the last pickup of it.
You want to try to go inside? Is that all right?
No, you weren't serious, right?
No. So can we just go a little bit left? So there's an overhang
Drop out about 10 meters
on the floor.
Keep an eye on that.
No, of course, what did you do
Yeah, we're
first?
just kind of getting set up here.
How about the slag?
Once we get you can see an even a little bit. Once we get a level, I'll give you a really good picture of it and see if we can.
I'm gonna drop
Yeah, there's some stuff over here.
My very far right camera there.
Your far right camera.
Right lower. See what's
Oh yeah, got some whip corals. Yeah, there's some stuff here we could take a close look at. All right. All right, I'm gonna zoom in. See what we got here on this wall. So, the whip coral. Lots of different soft corals. Cup corals.
It's a soft coral.
Soft coral, Gorgonians.
Yeah, lots of little cup corals, some maybe polychaete tubes.
Oh, another one of
Golden
those.
column across the gorgia.
These are beautiful.
Yeah, very nice.
Cup corals down there. If we settle down, there's a whip coral. So we've collected a few whip black corals, so unbranched black corals. They've all been in what we now call the genus Stichopathes. What we haven't yet seen is a coral from Sauropathes, which is another unbranched genus. And I think it's just here. I'll take a closer look.
He's saying 70%.
Okay.
So maybe, do you want to get a piece here? Yeah, okay, we need to get a raw sample.
So we're just going to look closely here. Do we see one row of polyps or multiple?
Jeremy, while you're looking at your black coral, I'm just going to comment here. It's Marty here, saying hi to Luke. Yes, we're all in the room here talking about the microbialite, and I've seen your message. So we've got that, and once Jeremy's had a look around for biological samples, we'll go in and try and get a sample of that microbialite.
It's good? Yeah, it's good.
Hey Ben,
Yo.
you don't need to stand out in the sun.
No, I'm hiding.
Okay, yeah, Bite has the camera zoomed in on that document too. So.
Okay. I'm hiding in the shade.
Okay, cool.
Okay.
Okay, it looks like another Stichopathes, so we'll pass on this specimen and we're going to get the rock sample. Do you know?
We got lots of time.
It's all yours. Where do you want to collect?
You go ahead and finish all your biology.
Okay, well, I'm not sure when the next question will be, so is it here?
Not quite right yet.
Okay, so we'll look around a little bit more. So there's a question about why they spiral. The answer is we have no idea. A lot of the spiral black corals tend to grow into currents, so it might be a way of the coral maximizing the nutrient intake by spiraling into a current rather than being straight. But then you also see contorted, straight, so there's lots of different morphologies. So I would guess that it's due to the abiotic conditions that kind of drive the shape of the unbranched corals. But we're not sure one way or the other. Some species might actually grow only in a certain way, whether it be spiral or contorted or straight. But that is a question that Marzia Bow is trying to answer, and hopefully with the assistance of our conserved elements, we're going to get to the bottom of that question. So we've got another soft coral here.
We're also much stabler though, so we just want to do it like definitely the James and then because we could actually come up with more lights and see the picture.
Okay. Yeah. Worst experience ever.
It's a good idea. Okay.
We probably got about 2 meters. You know what? Let me go on my arm and see how far.
Okay, we're going to stretch our arm and see how far we can reach. Bit of a stretch. Yeah, so there's a question. So yeah, the genetic analysis is going to help us solve our questions relating to comparing species to different morphological features, because it's one thing to identify a species based on its morphological features, but that doesn't tell you if it's the same species as another specimen that looks the same. So there could be two very different species on an evolutionary scale. So two drastically different species that look very similar. So we'd call that convergent evolution. And especially when a coral is so simple, there are no branches, one row of polyps, and all we have to go on are these little skeletal spines that, with features that can overlap between species. So how do you go about separating species that look so similar? Using molecular techniques that we're now using, we're using conserved elements for our analyses. We're able to sequence the DNA, and that gives us another line of evidence, so we can compare the morphological features to the molecular relationships between species, and that gives us multiple lines of evidence to say whether two similar looking specimens are one species, or if they're different species that just happen to look similar because the marine environment dictates that a coral will best survive with those morphological features.
I'll try to reach for the top stuff first. Just get the arm up there and see if I can touch it.
You're grabbing a rock? Do you want to grab the rock attached to the soft coral? Yeah. If it's all the same to you. Okay. You okay?
We're going to have to go for one of these.
Jeremy, would you mind just giving him the zoom as he goes in there?
Zoom in, yeah. Are you going down? Down here?
Yeah.
Okay. Oh, I thought you were going up. Yeah, no problem.
We were. It was just a little too far in my range.
Zoom out.
I might try to come on the bottom of the ramp or just like a little bit old trick in the supermarket where it's a pile of tins of cans stocked up. We could right on the road. So we walk
The bottom of it. Yeah. Oh, it's good right there. That's a really good one. Let's knock those off. Yeah, those over. That might be easier. Easier setup there.
I might sit on the porch on this guy. You see what I particularly like? You see right above the E there, which I dropped down? That little holes right there?
Well, nothing that's popping up. There's another piece right there. There's another one of these piece right there.
Actually, if I land out on the dirt here, can you check it out?
Okay. Is that still the stuff right there?
Zoom in.
Most of that stuff. Okay. That's even better right there.
Which one, left or the right?
I think I'm gonna go for that. Those two little nubs up here.
Yeah.
Just bang one out. That's gonna be sample.
That's right.
Almost. Yeah, we click behind you.
Okay, so it's Marty here. Just a little bit of an explanation about why we're flying around looking for just the right spot to take just the right rock sample. So when we were zoomed out before flying around, we saw that the texture on the outside surface of these limestone rocks looked a bit different, and we could sort of see this texture that looks a bit like cauliflower, and that suggested to us that this looks like it's a microbialite. Now, a microbialite is essentially a rock that's been precipitated by microbes. And this is of interest to us and a few of the people that we collaborate with. So we're going to take the opportunity to try and sample some of this material, and what we're going to do with it is, if we manage to get some, we're going to fix some in ethanol, and then we'll be able to look at that, analyse that as, you know, how it was when it was biologically alive on the surface, and we'll take an extra sample that we will then just treat like a rock that we will then just cut open and make a thin section out of, which is a really thin slice, 30 micron, that we'll be able to look at under the microscope, shine light through it, and have a look at the structures and textures that have been formed by these calcium microbes. And so that's why we're taking a moment here to collect this particular rock outcrop. So this is limestone. This is all part of a fossil reef that has then been colonised in the cavities and crevices by these calcium microbes that are precipitating more limestone.
One piece or so.
Right there, see how he sticks out there?
This one over there.
Joe, are you already trying it?
It's on Sunday.
As we zoom in, we could see small little corals, bryozoans. Cup corals. So it's obviously really hard calcium carbonate.
Hard stuff.
It's giving the metal claws a go. Where's the sledgehammer? Is that one?
That's what we need. Actually, that's when I was trying to yeah, that's this. Oh, it's too small.
What about this one? That'd be the right on the edge, right at the right laser.
This one right here.
The other one you tried, but just the very tip of it.
It's a good idea.
Get it?
It broke off. Slipped down.
Oh yeah.
There it is on the floor.
See it in the sink camera right there. Oh no, that's not there.
Isn't this it down here? No. Yeah. Can't be sure anyways.
Very, very small.
Yeah.
It's just a little piece.
Will that work?
That's nice.
Beauty.
Yeah.
Could be.
What's going up?
Beautiful.
Of course. Let's get back looking. What's falling on us?
So we have until like 5, is that right?
What's that?
We have until like 5.
Yeah.
So of course the empty bin.
Yeah, take your time. Sweet. We're going to drill at 130 metres.
Awesome.
So we can explore a bit along the face too.
Yeah.
Yeah.
Just going to rotate a little bit. Maybe core channel.
If I'm resting on the core channel.
Give it a try and I'll just push you away. Okay, ready? Yep, go.
Oh yeah, you're good.
I'm going to go for the future.
I'm going to porch it a little bit more.
Going for W.
Yeah, we're on the rails, so you're good.
This one, talking about the right now.
All right.
You beauty.
Beautiful. Pretty good. So just stick that in a bag of ethanol, 75.
Slide out of here.
Do you want another piece like that, or are you happy with it?
I'll take another piece of this easy bit.
Yeah.
Let's look for an easy bit. Oh, he knows where he's going. He didn't hesitate.
Okay, look, there's another one that's right next to that one. Right there. Oh, that's a nice one, right?
Just
Right up there. I think I have a restaurant on. Might push it down a little bit. Just crisp bits. Might have to go
All right. So we will meander. Look around for some interesting things to collect.
All right, Bridge, I got an updated waypoint.
Thank you.
Would you keep sliding laterally, or are you happy to crest over this?
You can go lateral. There seems to be a lot of stuff over here you can take a look at. What's with the light?
Just because because it overhangs so much, I'm going to come in a little bit more, but I just need to have an idea of what it looks like before I come in, because I can't see up here.
Yeah.
And if I bump in there, I'm going to break a light or something. So
Yeah. Is it hard to replace a light?
it comes out of your paycheck, so it's
Fair enough.
Well, let's have a look down in this cave here.
But you said your paycheck, right?
No, your paycheck.
Oh. Well, they won't be getting much.
Wow, look at that. Yeah, like this is a classic spur, but I mean, we're talking at 160 metres down. It's quite something. But what Adam was just talking about is, I mean, you're getting that view directly ahead of us, but there's clearly a lot of overhang, and we have to be really mindful because there's an umbilical cord between the ROV and the ship, and we don't want to either hit the ROV on the rock or get hung up with our cables. So we just have to be really, really careful here. Nice.
Seems to be a Gargonian whip here. Some more octocorals above. Maybe stalastrid there, potentially.
Yeah, it could be. So this is a zone we've found stalastrid corals in before. Again, they tend to be under these overhangs. They tend to be this delicate white coral. They're hard, like as in they're not a soft, pliable coral, and we've seen them quite regularly in this particular depth zone. So we're looking out for these fragile, delicate-looking white colonies, quite likely stalastrids.
Yeah, they're made of calcium carbonate, aren't they?
Yeah. Yeah, they have shallow water cousins, and but they have this quite typical is they're white in colour, even when alive, and sometimes with pink tips. They're quite a pretty coral to look at.
Okay, Luke, you're holding up there better?
It's easy
I'm just waiting for the ROV to get a bit more stable before I zoom in and out and pan left and right. It could be disorienting for the driver.
So what is it? When you say and they can film that okay. So you think this is an appropriate one? So you want to be framework column?
Sorry, just and these both cameras are looking straight up here. So this is on the wall.
Yeah, it's just starts to pick up.
Yeah, stuff with Marty.
Yeah, any of that stuff's really good.
That stuff is good.
Good stuff.
Oh, it's just overreach. Since better get it.
That'd be a good piece if you could break it off.
Oh, that is a good piece.
Nice.
So Sandra on Facebook is just asking, have we had minke whales visit us? The answer is no. This is the first. The Falkor has been in the Coral Sea for a while, but it's mainly been offshore Coral Sea out in the Queensland Plateau. So for this leg 2 of the expedition, we've been on the Ribbon Reefs. Now, Ribbon Reefs are quite well known amongst the whale watchers because of the minke whales, the dwarf minke whales that come here during winter. Now we're at the very tail end of winter here, so we think that those minkies typically appear around June, July, and have usually disappeared. I think by August, if I got my timing right, they disappear here in the Ribbon Reefs, and they're on the shelf. And there's quite a good tourism operations that allow you to swim with minkies, and there are a number of liveaboard dive boats that out of Port Douglas and Cairns in North Queensland that will take you out into the Ribbon Reefs and with a lot of guidance, and it's very well regulated, but people can go in the water hanging off slack lines at the back of the boat, and the minkies will swim up to you. And I've done this, and there's a few people on board here who have done this, and it's really an amazing experience. You ever done that, Jeremy? You swum with the minkies?
I have not.
Yeah, it's definitely a winter thing, but they've left this area now.
Yeah, we don't often
It's an
see.
awesome experience, isn't it?
Yeah, you've done it?
Yeah, it is like, if you know, you know kind of thing.
Yeah.
I was like astounded actually, quite how incredible it is. Beats whale sharks any day.
So on board we have a really international team, including with the crew, of course, from all over the world, but we also have Nelson, who works at CSIRO in Hobart with the Marine National Facility. He's part of the geophysical survey and mapping team, so it's very good to have Nelson on board. Nelson's been recruited for this particular expedition because of the difficulties of getting people in and around from different parts of the world to join the ship. And Nelson's a big part of the multi-beam mapping team on board, so it's great to have Nelson here from Geophysical Survey and Mapping from CSIRO.
Yeah, that's Stu. Stu will be stoked on that.
But Nelson's a Queensland boy too, and he's also swum with the minke whales, and it is it is a great experience because these minkies are really curious. They're not just passing by; they actually come up to you. And I've got within a few metres of them, and that's not swimming after them. You just sit there quietly off the back of the boat in the water, and they will swim right up to you. And they're quite big animals, anywhere between. They're kind of what's classified as a small cetacean, so they can be, up to about 5-6 metres long at the most. But I've had them 4 metres or longer swim up to me within a few metres, and they keep coming past. It's not just one pass; they'll just hang around all day. They just seem to be very curious about humans.
Can I zoom in on this coral here?
I'll just hold it steady here.
Okay. This might be interesting.
What have we got here, Jeremy?
If it's a black coral, could be Myropathes. Seems like it is. Get a little closer. We can get pretty close here. Or is it a octocoral? So what we're looking for is the polyps. I think those are black coral polyps with 6 tentacles. How would you go about collecting one of these branches? Zoomed out. If it's too difficult, it's okay. It's kind of in the middle of the cave.
I might be able to, if you want a free flight in there.
2.5 hours on this.
Just come up with a shield like last time.
Yeah, you could just grab a branch on the way, and then you pop straight up if you want. You know, instead of
If you get the whole thing, it's fine.
it's okay to get just one of the branches.
And then if we get the whole thing on accident,
Yeah.
Okay.
Sure.
Just in case.
Yeah, of course.
Yeah. So they do it. You guys must come up from used to it. Just get in there.
Look at the sonar. See how it goes right in?
Yeah.
Just go for a reach there.
Imagine if you had a little ROV that could detach from this ROV, can get inside, spin around, and go back home.
Yeah.
Kind of like a Roomba.
Well, this is that's how they explored the Titanic, isn't it? They send little Jason off from the main ROV.
That'd be fantastic. So we're trying to reach into this little cave opening to grab a coral. Now, it's hard to say what type of coral it is without getting a closer look, but regardless, it kind of seems different than, it doesn't look like the other soft corals in the area. If it's a black coral, it's probably a myropathid, because we're at 150 meters, so it's fairly shallow for black corals, which means there's 3 families that can occur at this depth. And you can see it's got pinnulate branches. There we go. We got a coral.
Yeah, good colony.
Looks like a decent size one.
It's a pretty big coral.
That kind of thing,
Yeah,
Yeah.
size.
Can we zoom in while we're holding, or if you want to move back?
Yeah.
Yeah, actually.
Slide out of here.
Yeah.
Yeah, I got a bigger one.
Lots of branches here.
Sorry, I didn't mean to grab the loop.
Oh, it's all right. Sometimes it just happens.
I'm trying to twist it off, but
Yeah. Totally. We have the basal attachment, which is nice. So we have all the information we need to identify the species.
it's hard. Definitely hard.
It's hard. You mean black?
Hard black.
Hard black coral. There are soft black corals. All right, I'm gonna zoom in. Is that all right?
What description for this, Jeremy?
Myropathid.
Quick reach. That little bit you did. Oh.
All right, so yeah, you can see the tentacles. You can see it's not an octocoral. You can see that it has few tentacles per grouping, surrounding the feeding mouth or polyp. So it's certainly a black coral. And
so, Jeremy, you're saying each one of these little dots is a polyp?
So if you look close, I'll get as close as I can. So you can see the tentacles. So each pair of tentacles.
Yeah.
So those are connected by one mouth. So if you're looking deep into this image, you can see it facing us. It's easier when you can see the. I'll try to get an image. Yeah, I'm gonna try to get an image of the polyp facing towards us, and then you can see how the tentacles are arranged around the polyp or feeding mouth. This might be a good one. They're still all facing away. So we're looking at the back. Can you spin your hand around?
Yep.
Microscopic. There's a lot to work with here, isn't there? But yeah, that'll do. All right. It's amazing we're able to look at this in the water. So we're going to look closely.
I'm trying to get in more light for you, Jeremy.
Okay. If we could focus here.
Start focusing on getting more light for you.
Am I in manual or automatic?
You're in auto right now.
Okay. That's probably for the best. Yeah.
There you go. Try that now.
Okay. We're going to zoom through this foreground into the background. And here we go. So that's a polyp.
Yeah.
And you can see the tentacles around the outside. It is a better view. So you can see 1, 2, 3, 4, and then it's harder to see. So 1, 2, 3, 4, 5, 6.
Yeah.
So you can see these polyps are elongated. So instead of the tentacles being all surrounded around the mouth, feeding mouth, they're a bit extended.
Okay, and so they have a pair. You'd say a pair of tentacles per mouth?
There'll be 6 tentacles per mouth. So this is the mouth.
Yeah.
This is a pair. This is another pair. This is the only mouth surrounded by these 6 tentacles.
Right. It's spread further along the branch than I expected.
Right, and the different families will have different arrangements of. Here's a good view. So you can see that's a mouth, and that's a mouth. In between these 4 tentacles, these 2 go to this one, these 2 go to this one.
Yes.
1, 2, 3, 4. You can't really see it, but there's 2 there, 5, 6.
Yeah.
And
okay, that's quite clear.
You can see along see a mouth. A mouth.
So the round dots are the mouths, and you've got. It's quite strange how they've got the tentacles like that.
Yeah. All right, let's get it in. I'm going to say 1A.
1A? Same way right now. But.
There you go.
Wait,
yeah,
let me get this straight. You wanted us to fly in a cave to get this, and now you want to do 1A?
I'm the one with one pinwheel, one little branch.
Yeah, we blamed it on cookies. Beautiful. Let's get her. It's pretty big.
Yeah. I mean, do you want to rip it in half with both arms and take?
Yeah, we can take a snippet of it. Get it.
All right. I don't doubt your abilities.
It's pretty big. I want to keep it like this.
It's going to hang nicely in our wet lab.
Matt, I'm going to take a picture of it too.
Do you want to snip it and put it in the kitchen?
You can't lay it in?
Yeah.
That's right.
Actually, there should be a way to get a pointer finger on the screen.
Do you want to leave 4 closed and jam it in the tree? Just something about that.
I don't think these things
Tie it to the roof.
Roof racks, yeah.
It's like a Christmas tree.
What's that? Harry and the Andersons?
Yeah.
Tie it to the roof.
Yeah, if you get it half in, it's fine. There you go.
Fully in.
There we go. Hey, this is a big bio box.
And 1 branch.
So the base of the coral is quite important for you too, isn't it, Jeremy?
Yep. So right above the basal plate, which is what we're looking at now, some species have what we call striatum. Striatum are these little lines that are found on some species and not on others. We don't yet know if that feature is informative in identifying and separating species. The only way to figure that out is to determine whether a specific colony does or doesn't have it, and then sequence it, and then put it in a phylogenetic tree. Then you look at the tree, you look at the tips of the tree, and if certain clades all have those striatum and other clades don't have those striatum, then you could tell that it's certainly a morphological feature that represents similar, it's a feature that's similar among closely related species. Otherwise, it's a random feature that doesn't help us identify the species. And that's the same with pinnulation pattern. So you can see this coral has pinnules. Pinnules are repeated branches along a branch. And if you look at the phylogenetic tree, large clades, entire families are either completely pinnulate species or completely non-pinnulate. And what that means is it's a feature that is evolved within these different families or not evolved. And so that's, so if you have a kind of a key of how to identify a species, the first thing you'd say is, well, is it pinnulate or not? Yes? Okay, then it can only be these families. So it's a useful feature to help understand the taxonomy and the evolutionary history of the group.
But the DNA sounds critical here. Like without the DNA, you can't really separate them out at that sort of almost independent understanding of where they sit on that tree.
Yeah, you're taking lots of leaps if you don't have that second line of evidence. You know, whether two species are closely related because they look similar. Well, you know, you can only guess at whether that's true or not, right?
Yeah.
So they could look similar because the current and the temperature and the sunlight dictates that morphology is what that coral needs to survive. And they could have evolved separately but look very similar.
Yeah.
But the DNA would tell us whether that is a result of them being similar, closely related with small mutations between the species, or if they're completely different and just happen to look similar. Oh, great colony. So just letting everyone know, we now have 6 out of the 7 black coral families that have ever been described. That's a pretty big feat, I'd say.
From this voyage?
In this voyage, yeah. We're missing just one family. We're missing Leopathidae, and that's a family that we really do expect to find at these depths. I'm surprised we haven't come across it yet. On the last expedition that you were narrating, we saw lots of Leopathidae, so we know it's around. I'm just surprised we haven't seen it yet.
Yeah. Or is it only in the offshore Coral Sea, or you expect to see it on the Great Barrier Reef here?
I don't know. Yeah, I'm not sure. We'll know soon enough. All right, so we're looking. Oh, wait, hold on. Can we hover right here for a second? I think we're seeing the first zooxanthellae hard corals.
Oh, okay, that's interesting. 140 meters.
Actually, this is about the deepest that we found the other day, where we found some Leptoceres.
Yeah. So this is this brown disc-like shape. So this could be our first Leptoceres. This is a scleractinian hard coral. This is exactly what Tom Bridge, our mesophotic coral expert, is interested in. Let's have a closer look at that. Or it could be just a sponge.
Maybe not. Yeah, no, it's not.
I don't see any ridging that you get from Leptoceres, but it's about the same colour.
Right.
All right, so we keep looking. We expect to see that fairly shortly because we're 140 metres. Do you remember what depth we were in?
Like 120 yesterday.
So, you know, not far away, literally 20, maybe 20 more metres up.
Yeah.
We'll just see. So, not Leptoceres, but it'll be a very similar sort of colour and a similar sort of shape.
Yeah. Just more features. You also have the central column polyp mouth.
Can you keep rolling there?
Yeah. Yeah.
Definitely getting more colour coming in, aren't we, Jeremy? There's lots of, this red coralline algae is becoming, it's starting to give a lot of colour to the rock.
Right.
And we're getting more of this green algae. We saw that yesterday.
Right. So we're about to see a pretty big shift in the assemblages of life here. As we go from light not really penetrating to corals that require light to survive.
The other big thing we're interested in is about 120 metres, which is where we think low stand sea level was back in the last ice age, about 20,000 years ago. We talked about this extensively yesterday with Jodie Webster. But if you go, so just repeating, if you go back 20,000 years, last ice age, globally the sea levels have dropped around about 100 metres. So we might see some evidence of wave cutting platforms. Cave development is probably what we're going to see.
Armored fish.
Sure. I know Cody will find one up here.
Some nice feather stars, Comanche with feather stars just attached to that coral.
Between 130 and 120.
Oh, 130 and 120.
Between 130.
It's Gemma. It's Gemma. Don't grab it.
Seeing lots of soft corals.
Bridge.
Go ahead, Luke.
Hello. Is it going to work for you guys if we come over to port slightly, maybe 10, 15 degrees?
Jeremy, we're going to get Gemma to join you too because we're starting to see fish.
Yeah, it should be okay. But
We're useless.
You're not useless.
So if you want to come to about here?
Yeah.
Started a list of everyone we've been seeing.
Ah, very good. Welcome back, Gemma.
Hello, team.
You want to just introduce yourself, Gemma, if anyone else?
Yeah, let's slow down a little bit.
Hi, everyone out there. This is Gemma Galbraith from JCU. I'm going to be joining Jeremy as we come up into the kind of middle of the mesophotic. Going to be keeping an eye out for some of the fish we're starting to see. Some familiar, some not so familiar, so hoping for some interesting things. Last collection was
Pretty good? Yeah, pretty good.
real nice. So many crevices,
Yeah.
stuff to hide in.
Yeah, so we're seeing lots of soft corals here. Feather stars. What I assume are sponges.
Some sort of solitary ascidians.
Oh yeah, that's probably more like what they are.
Yeah. I would definitely start expecting to see some holocentrids, squirrelfish, soldierfish, red, big eyes. Love these kind of overhangs and holes to hide in.
Yes, fish.
Fish.
So we're looking for a rock, is that right? A rock of sorts?
It's got to be just the right one, Jeremy.
Yeah, the right sort of rock.
Oh, it's just a little chromis, I think, down in the left hand side. I didn't get a great look at that.
Yeah, there's nice area. It unfortunately goes as far as the Falkor.
We're looking for a particular kind of rock.
I assume so. All right, because this is the depth at which used to be sea level, when the sea level was lower than it is now. So getting insight to
Make a geologist of you yet.
yeah, what the coral reef used to look like before the sea level rose. How did I do?
Pretty good.
Very good.
Very good.
Thank
If
you.
you want to sound really legit, call it paleo sea level.
Paleo sea level.
Yeah, geologists say paleo.
Is that always this depth? This is paleo
At the
sea level.
lowest point of low of
All right.
That's
So we're
20,000 years ago.
20,000 years ago.
When all the water was caught up in ice caps.
Very good job.
So we're trying to balance on, we're hovering, trying to get close to grab one of these paleo sea level rocks.
There you go.
Thank you. Thank you. That's pretty good.
Barely on there. Okay. Beautiful. Yeah.
This is 3 at 5, is it? Huh.
385.
Yeah.
This is dive number 385 for those of you who are keeping count. We're at 130 meters and it's 23.3 degrees Celsius. As Rob would say, this is swimming temperature, but not for everyone.
It's warmer in the water than it is in the air.
It's pretty warm. Still not warm enough. I do well in 29, 30. That's my kind of temperature.
Tropical kind of guy.
Yeah, Philippine temperature.
Alright,
PNG temperature.
I like it.
So Jem and I worked in Papua New Guinea together, and we discovered the new genus of black coral, Blastopathes. Jumping off the same boat, I remember doing a dive, assisting another colleague while rolling up some transect tape, and I saw out of the corner of my eye this big branching, elegant coral over 1 meter tall. So I dropped the transect tape, swam down, and then just stood in awe, looking at this, undescribed genus of black coral. So then later on, I was able to bring it up to the surface, and then I handed the coral to Gemma, and I had my camera in the other hand, and I took a picture of myself handing it to her, and that's the picture that I always use when I try to share with people the excitement of finding a new genus.
Quite an amazing moment. I think we've also got our boat driver Jerry in the background, kind of pointing at it in shock and awe that you emerge with this.
It was a really exciting moment. You could see in everyone's face.
Yeah, and the water up there is what, 30, 31 degrees? It's like a bath.
It's like a bath, yeah. You don't wear a wetsuit. You just jump in with your BCD, and you come out of the water after an hour and you're still warm. It's fantastic. That's how you dive.
So we're totally ruined now.
Yeah.
We can never go into
Who would want to?
sub 30 degree water again.
No, absolutely. So we're in a location here. We're about to grab a rock, and I see right in front of me a coral that I might be interested in, but I'm not going to zoom in until they're done doing.
Wait till we have the rock.
Yeah.
Glad to see you guys. Great Barrier Reef.
The rocks come first.
The rock's been waiting 20,000 years.
Also, you've got 5 geologists.
Yeah, we're outnumbered here.
We are outnumbered, yeah. This is an ambitious rock. Oh, yeah. Excellent.
We're going to put an inset.
Full picture if you want.
Yeah.
Yeah, just a full one.
Yeah, just briefly, just to show people. Just going to put.
Oh wow!
This is the back of the ship, and you can see Ribbon Reef 4 in the far distance. It's about 1 kilometre away, and you can see the Australian mainland in the far distance. It just shows how beautiful and calm it is today.
We've been really lucky on this trip. This has been the weather more or less for the last week and a bit, however long we've been on the ship for. And there just seems to be good weather for the rest of the trip as well. The forecasts seem to predict. So we've been really lucky. I think that's Beaufort 1. Is that right? I know Beaufort 2 is when there's white caps and there aren't, or is that 3?
Probably 3.
Alright.
Slight.
Sometimes you forget you're on a ship. You know, it's not even rocking. It's like we're cheating. Can I just? Are you getting something else now?
Yeah.
Okay, go for it.
Where's that coral you want?
It's like right in, I just want to zoom in on this orange, but we can do it after.
I'll grab this real quick.
I will allow it.
Thank you, Jeremy.
I'll even let you put it in one of my boxes.
Tell you
wow.
who runs the show here, Jeremy.
That's right.
Oh, it's going to be a lot of cool life actually on these rock samples coming up.
Yeah, I bet that's a stylastrid. That little, if it's hard.
Okay. Looks to be some maybe encrusting sponges and perhaps a couple of obsidians. It really is so great when all that stuff comes back up, and you know you could pore over it for ages trying to look at everything.
Oh yeah, and there's life that we, even with this camera, we can't see. But every rock that comes up, there's tons of stuff on it.
Yeah. Oh yeah.
There's life on life on life.
Good ground.
Yep, that's great. Thank you.
Yes.
It's all yours, Jeremy.
Thanks.
Just gonna take a look at a coral with Jeremy. Kind of looking right
Interesting. Looks like an octocoral. Alright, I was hoping it was something different. After all that, nothing.
Force me with me.
J-Rod.
¿Qué pasó, mi amigo?
The colours when you zoom right in on that rock with everything dotted all over it is pretty cool.
It's quite spectacular. Let's do it again.
Yeah, I like it. It's like some modern art canvas. Yeah, look at that. Yeah, it's just crawling with life.
Oh.
Oh.
Ooh.
What did you just find?
Ooh, okay. This is interesting. Thanks. So yeah, that last coral we looked at was soft coral. This is a black coral. I thought they'd be the same, but So you can see the fewer tentacles.
Oh, little nudie breakdown.
Oh, lovely. Well spotted.
Lovely one.
That is a beauty.
That
Can you
is
zoom in on
gorgeous.
that?
Yeah, I just thought it was
It's
Oh,
fairly
lovely.
active as well. on the
Well
move.
spotted. Oh, let's get a look at that one, Jeremy.
Almost,
Oh, look at the colours.
almost lost in the kaleidoscope of other.
That's as deep as we can go.
Ah,
That's pretty good.
that's a view.
Ah, spot on.
Beautiful.
Well done.
Who can identify this nudie?
That is.
There's so many. I've
slightly
seen books
beyond my.
like
oh,
an
yeah.
inch thick on nudie, different types of nudie branks. There's so many of them. That's beautiful. Yeah. This nudie brank means naked
There's a black
branch.
coral there.
Like these. And they have some really unusual ways of defending themselves. They can actually eat soft corals and incorporate the poison, the toxins from their prey, even the nematocysts from some of the soft corals into their own tissue and sting predators that might want to eat them. So they're using the venom and stinging nematocysts from those soft corals in their own flesh, which is pretty amazing.
All right, it's all yours.
These little bumps, these little nodules are quite interesting, aren't they?
Seems
What do
to
you
be
think, Marty? What are
a.
these little fine-scale nodules?
Yeah, I think it's that
just-
microbial texture that we were
Yeah.
looking at earlier.
Okay. Yeah, it's good to have got some experts there looking in on our nudies. That's great.
Oh, I'm sure these guys love looking at nudies. Everyone does. Everyone goes through a phase where they have that book and they're just flipping through it after a dive, looking for it and have a checklist marking off the pages when they've seen them all.
You see the light down through some of these cavities. This is really this huge Swiss cheese effect right through the face of this rock wall here.
So the interest in this coral here, it doesn't seem to have, so I was discussing before whether species can be pinnulate or non-pinnulate. It doesn't seem like it has pinnules. I could be wrong though, it's hard to see from this angle, but it's a fan coral. Yeah, the branches seem too there isn't much order to the branches, so it seems more like they're branches rather than pinnules, which would make it in the family Antipathidae. So Antipathidae, none of them have pinnules. Myropathidae, they all have pinnules. And those are the two main shallow water families. And we've been collecting lots of Antipathidae branching in 3 dimensions, so 3-dimensional branching colonies, but this is a planar colony. So this would be our first planar Antipathidae specimen. And it's from 132 meters, which is a bit deeper than scuba divers can go to grab species like this. So I assume it's a specimen that isn't often collected. And if it is, it's very deep. It's probably as deep as this species goes.
Looking for another rock specimen about 110 meters.
Sounds good.
The Calamita floating past.
Two? Yeah, 2. I mean, it's pretty small, isn't it? What do we have in 1A?
Rocks.
Throw on top of 1A rocks? Yeah. Now that I look closer, it probably looks more like a myropathid, which is still interesting. So you could see that these branches tend to occur in some sort of a distinct pattern along previous branches. You probably let go. It won't. It's going to be attached at many points.
Oh yeah, it looks like you got.
Yeah, so we have Lisa who's going to share the nudibranch image with the Twitter worlds, the Twitterverse. And as she does, she has the world responding to her messages very quickly. So I assume she will get an ID before we leave the site. You'll probably rip off the branches before you remove it from the basal attachment.
Alright.
We could, you know what? We could leave it. It's okay. It's a bit hard to get though. You're there. You're in there. Okay. Do we have a ID on the nudie?
Putting there a couple of ideas. Got Bulgaria, Maculata. Based on it having spots rather than
That's okay.
So I got
kind of continuous colour.
part of it out, then the other.
I'll just take a zoom in, just see how much of it we got. I mean, yeah, this is enough. Do we have the basal attachment point? Is that the attachment?
Yeah, I think so.
All right, coming close. Coming close.
Work on the same principle as lefty loosey, righty tighty.
Actually reminds me
Because you
of...
got that wrong yesterday.
I know, I'm really not a builder. We built a calibration cube yesterday and I would get it right like 15 times, then the 16th nut I would do it backwards. Yeah, okay,
Yeah,
that's good. Go for it.
Can you focus that, Jeremy, and I'll get a different grab on it?
I'm letting J-Rod do it. But this does remind me a lot of using a scanning electron microscope. So it's the exact same setup where you have a coarse and fine focus and it's just knobs that you need to spin one way or the other. Oh, looks great. Oh, you know what? Yeah, it's interesting. I can't. So, I mean, it's
Got some pretty irregular branching going on.
It's pretty irregular, yeah.
Pretty odd.
So does that mean that it's non-pinnulate? But it has so many that I think we would consider it a pinnulate, first order, and then sometimes second order pinnulate.
Okay.
That's kind of what we're going. It's really hard to tell. It's the same with the deeper species. It has to be completely perfect pattern for it to be pinnulate. If it's missing just a few, it's then considered branches, and it really throws off the taxonomy. It's a tough group to work on. But yeah, that's a great picture.
Nice.
Screenshot, yeah, you got that?
Yeah.
Let's throw it in the box.
Get it in the box.
Which one?
1A, please.
It's already open for you.
All right, so then after this, we're gonna go a little shallower and get a rock sample at 120 meters?
110.
110 meters. That's a nice little colony.
Jeremy, what did you think that was again? Or do you want me to-
Call it a planar black coral. Yeah, sometimes these black corals like to hold on they have little rigid branches that get stuck in the metal arm and don't let go. There we go.
Nice.
All right.
Onwards and upwards.
And we're going to look for some fish. Because we've got Gemma here. Alright, we got Rock'em and Sock'em being pulled back in. That's what we call our bumpers. Rock'em and Sock'em. Huh? I thought right was Rock'em.
Sure, that used to be hard.
No, we changed it to
Did you change it?
Yeah.
Oh,
Do you remember?
Rock'em and Sock'em and the bumpers, but there's disagreement as to which is which.
Yeah. Well, originally it was left as Rock'em, right as Sock'em, but I thought you want the alliteration. You want the right to be the Rock'em, left to be Sock'em. But I guess we're still deciding.
No, Adam was the one that named it, so if he changed it, I mean, he can be like Bert and just keep changing it.
Okay.
Shout out to Don Cherry for the rocket soccer buttons.
There you go.
So we'll just flip the, we've got the sit cam and the science cam.
Nice.
Just flip it back there. That's the sit cam, just to show how steep it is.
It's a pretty vertical wall, isn't it?
Yeah.
Bridge ROV.
Wow, we.
ROV bridge.
So
Moved you to the west a little bit. Alright, west, next.
You realize that's a wall right there, right? There's a wall right there.
Wow. Yeah.
Here's our spur and grooves again, but they're like 100 meters high.
Here's the launch point.
Alright. As long as it doesn't come in, before it was coming in.
Here, I can actually change it a little bit. Alright, update.
A little bit better.
So we're seeing lots of soft corals,
Oh, yeah.
sponges.
Wow, really eroded. Look how. Yeah, lots of cavities. You can see light getting in right at the back there, so very, very hollow.
You know, oh, Rob, I think you'd be proud of me. Yesterday, when we got one of those sediment samples, they wanted to know what the texture was, and they said that you have to put on your lip and feel the texture on your lip. And
Now you chew on it. You chew on the mud. See if it's silty.
where's Marty?
What marine geologists do. No,
Marty just had me put it on like lipstick. I'm feeling it all.
You're the one who put it on like lipstick. We just haven't.
no, we chew it. We don't use it like lipstick.
Gosh.
Oh,
Yeah.
look at this. It's nice. Big soft corals.
Yeah, big sea fans.
Is that a nudibranch there? Is that right?
This might be a good place us to look at for a second.
Pretty large.
You want to do a perpendicular look through the sea fan? That's probably hard to do, right? Is the
Oh, yeah. Got a few chromies over there. Yeah.
Just down here.
Reef chromis.
zoom in?
It's a little damsel. Is that a damsel?
Yeah. It's like chromis alpha tends to be quite deep.
No, that's
sponge.
Yeah, sponge with some
Oh, yeah.
things on it.
More life upon life.
Yeah. Sea fan is beautiful. Always looking for seahorses, of course. Although I mean, what color is this? Brown, yellow?
Orangey colour. Yeah. It'll be, if it's there, it'll be exactly the same colour, but you'll just see a frond moving
there's some brittle stars on it.
Let's look for a minute. Maybe we'll find something of interest.
We can see a shrimp right moving in there on the main branch.
Huh?
You can look where I know you want.
Okay, thanks. I don't think anyone's home here. There's a lot to look at. Oh. A little shrimp.
There's a little thing dangling up there. See it right there? Right in the back? Up to the right. Right there.
Yeah, something's
Wait a
waving.
second. We got a pygmy seahorse.
Yep, definitely. See the coloration?
We got it.
See the tail hanging on the...
All right.
Yeah, you can see the tail.
Just around the
wrapped
other,
around the
other
front.
side.
All right, we're gonna try to. This is.
Yeah.
Oh, here we go. All right.
Well spotted, Jay.
Yeah, this is all J-Rod.
Yeah.
In the middle.
Yeah, see his head's coming over. He's on the back.
Pink and white stripes.
And he's got little bumps that are the same colour as the polyps.
Yeah.
And see his eyes? He's got a little black eye.
The thing about these pygmy seahorses, if you go to the other side of the sea fan, he'll just turn away from you.
Yeah, he's looking at us.
So this is the best view we'll probably get.
Yeah. So he's got his tail wrapped around it, and his eyes just poking through.
Yeah. We've been saying we've wanted to find a pygmy seahorse since we started this survey, and I'd like to know if any other ROVs have ever spotted a pygmy seahorse.
Size-wise, we're talking tiny, maybe 5 millimeters, centimeter, or maybe a centimeter.
Yeah, sort of like the size of your fingernail.
Yeah. Seriously? Yeah, tiny. That's why you really have to look closely, looking for something that's moving. So, J-Rod, our leader of the ROV team, just saw something bobbing behind, and of course it's quite clear now. But you've got the tail; it's actually wrapped, hanging onto the branch, and you just see his black eye, a bit of a googly eye, poking through the gap between the polyps there.
And seahorses, they live in pairs. So there's a chance that there's two or pairs of seahorses on this sea fan. So should we zoom out and look for another? No.
Let's just enjoy this.
Okay.
Yeah. You've been greedy now.
I'm sure there'll be more.
Okay, so it's bending away from us. So, we're looking from the tail view.
You want to go to the other side?
It'll spin up. It'll spin away from us, I think.
Probably going in the current.
We could try. It's not going to go to another. It's not going to swim away. I'll tell you that much.
Got some encouragement on the chat to go to the other side.
Yeah.
Easier said than done.
Yeah.
We're on a vertical wall at 100 meters, 120 meters, and
let's see what it does.
That's it's bending over. It's definitely seeing us. It's looking at us now, right? We just got quite a good perspective of it. Now it's underneath. I find the coloration amazing because there's little bumps on its body, the same color as the polyps on the Gorgonia.
Has anyone identified the species? Is it Denise?
Seahorse experts.
There's a Denise. There's
Denise. Yeah. What's the genus? It's not Hippocampus. Is it?
Yeah.
Hippocampus denise.
Oh, here we go. We've got some suggestions coming through from Gwen.
Yeah. Okay, we've got hippocampus. Yeah, I was on a paper that identified a pygmy seahorse from Osprey Reef, and it was a Denisi, and it was about this depth. It was with Dougal Lindsay, actually. Dougal is our plankton expert. He's in Japan, and he brought his Picasa ROV out to the Osprey Reef, and we were able to sit down and look at these gorgonians. We wrote a paper about it. But yeah, still very, very rare. Very hard to see, harder to find. Very lucky here.
Yeah.
Scuba divers can spend hours crawling around gorgonians looking for these guys.
You know, but this is really beyond diving depth, so it's very hard to find these. We've just zoomed out a little bit to look for any other movement because, as Jeremy said, they're often in pairs. And we're just looking for anything, you know.
Now that it's focused, you can see it, you know, from far away. It's the essential.
Yeah. Amazing that you saw it though, Joe. I mean, look at the size of the fan and the size of the seahorse. You know, we're talking maybe 5 millimeters, centimeter at the most.
Wow.
Yeah. Tiny little things.
So maybe we could look for the tail on other branches because the tail kind of is a white striped tail.
Yeah.
Glenn is saying maybe the campus Denise.
Yep. That was my first guess.
Yeah.
Possible it could be on the one on the other sea fan that was like.
It looked a different color. Yeah.
Wow. Like,
you want to give it one try?
On that.
Look on the other side.
Fine.
Of course I do. You ready?
Yes. Let's do it.
Yeah. Yeah, we agree. That's probably the find of this ROV transect. Yeah.
Certainly.
Okay.
Oh, they look similar in color, maybe. A little bit more now. Oh, there's lots of this type of sea fan. So there could be, you know, many seahorses in this area. I mean, all they need is this species of gorgonian to live, and they live within this colony until, for whatever reason, this colony might die. Then they'd have to swim looking for another one. But otherwise, they can't hide. They're pretty defensiveless.
Just trying to reposition the ROV here, but of course it's tricky.
Let me know when I should.
Very steep.
Zoom in.
Just trying to rotate around just a little bit more.
Yep. Yeah. This looks like something you'd see as you're diving a wall, you know? Just a little bit deeper. We're gonna try to catch its face.
Here we go. Here we go.
Oh yeah.
Oh, perfect. Well done. Well done. You can just see his little pelvic. Is it like they're? Are they pectoral fins? Like what are we seeing there? They're Gemma.
So
Look at that.
obviously, as a seahorse shape, this is quite a different morphology to other fish, but really they've got all the same features, just in different places. So yeah, Rob's right. We're just seeing little flutters of his pectoral friends, probably up near gills, and turning away.
Yeah, there's a bit of current thrown at it, so it's being knocked around a little bit.
That camouflage is just amazing.
Yeah. But it's quite symmetrical, isn't it? You've got the buds, I guess, exactly the same either side.
Right.
Yeah.
Which resemble polyps.
Yeah. It's looking at us now. There we go. Oh, so
Wow.
you can just see his gills. You know, the gills flapping away.
Just beautiful.
That's quite incredible, isn't it?
I'm thinking we grab a piece of the coral because we have an image of the seahorse. If we have a piece of the colony, we can then identify the species that associates with it. And it doesn't have to be. It doesn't have to be of this colony. It could be the one next to it. We could just see if it looks the same.
I think we should leave this this particular fan coral hole alone. There you go. Hey.
What a life, hanging out at 120 metres on that gorgeous sea fern. Not too shabby.
No, don't see any other mate.
Lonely seahorse. Should we pan to the sea fan next to it?
Yeah.
Oh, you want to get closer? Okay. Yeah, sure. Let's try to get. Oh, it's moving.
It's moving.
Well,
Yeah.
hey. Oh,
There we go. Grab that. Just see its tail. Just hook onto the next bit.
amazing.
Oh yeah. Yeah, it's as close as I can zoom in.
Yeah, I'll hop over just in that step.
Yep. Is that good? You need a tiny saddle. There are actually a lot of corals in this area.
Oh, hang on.
Bit of turbulence here. Still there.
Yeah. We may have to leave him.
Yeah. Why don't we?
I'm just drifting off right now.
Let's look at the. That's right. That was fantastic. So the coral next to it, I think they look the same. We could just take a zoomed in look and grab a piece. What do you guys think?
Looks kind of different.
Looks different.
Yeah.
All right. I call it different. All right, we can keep moving on. That was nice. It was an interesting area. Yeah. So we're 7 meters away from where we'll take our next rock sample.
good. That was the find of the day.
Oh yeah.
Yeah.
Straight up. Just I was looking for mine, they'll go back there and just straight up.
We should have had the Schmidt Ocean Arm, the logo go into the back of the image. You know?
If advertising, yeah.
Product placement. Alright.
Just let nature be beautiful.
Oh, we've got a little
Okay.
hogfish
I
down
could also
there.
Photoshop it in after.
This is a type of wrasse.
Watermark that.
Just disappeared into a hole. Diana? Diana's hogfish. Let's check.
Hey, Eric, I'm going into rescue alright.
I'll have to on YouTube.
Yeah, these sea fans look a bit different.
Interesting sponge at the bottom of this, this big fan coral.
Control deck.
Go ahead there, Deck.
Hey, Chris, I'm just going to engage your pinch wheel here. Starting to go a little slack. Yeah, 178. If you want to pay out, just give me a holler, okay?
Roger.
Okay, pinch wheel's engaged.
Back empty.
If you want to get in closer, if that's larger, keep going on.
That's okay. We can go.
It was 110, correct? Not 115?
Yeah, 110.
You're gonna like the way this looks.
So the sea.
Whoa, look at this one.
Wow.
Now look for the tails. Did we come across this before? There was a really interesting sponge at the bottom.
That's a different one.
Is it? Okay.
There's like a haze over the
Yeah,
coral.
it's like a like a film, like a membrane.
Yeah.
That little holes punched in it. So.
That is odd. Yeah.
Oh, they're naked branches. The polyps are either in or gone. I guess
Oh, so it's
with,
just the lack of tissue.
potentially
The
some
polyps.
grazing nudibranchs or
Yeah,
things that come
okay.
and
I've seen some brittle stars.
munch.
That spider right there?
Yeah.
Ooh,
The little white and black thing? Right.
is that a pycnogonid or is that a
Oh wow.
or is it a brittle star?
I got you. Move you along.
How is that?
2, 3, 4.
10.
Because we do have pycnogonids out here. You know, they're sea spiders. They're related to arachnids. Or is it just another crustacean? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's a decapod. No, it's a crustacean. A decapod of some kind.
Shrimp.
Yeah. We've got 10 legs here. It's more likely some kind of strange crustacean.
So we're just looking down for a second.
There's a brittle star down
See
in the
this
corner.
black coral right here? This branching.
Okay. The white one? No, the little guy. Come back.
This guy?
Just keep your zoom right there.
Keep your zoom in.
Yeah, just keep right there. I'll come back in.
Okay.
And J-Rod, this would also be another good spot for a rock if we can find one.
In this spot right here, or just anywhere around here?
Well, 110 metres, just need a good solid
I think around the corner there's going to be a better spot.
Yeah, okay.
Yeah, if you could pluck, so this is in the family Antipathidae. So this is, I mean, they can be found this deep. We're at 110 meters. It's a simply branched colony. It's got one stem and then branching on usually just one side or the other, not on both sides in one location. And it branches to the third order. Be interesting. So the family Antipathidae has more species than any other family, and it's the taxonomic dumping ground family. So if you don't know where a species or a genus belongs, you throw it in Antipathidae and call it a day. But now that we have molecular techniques that allow us to really understand differences between species molecularly, all we need now is fresh tissue to sequence, and we can place it either in a different family or in the proper genus. And this is a really interesting depth to survey at because it's too deep for a diver to go. But a lot of places, the deep have been explored really well because ROVs have been built. They can go all the way down to 3,000 or 4,000 meters. So rarely do ROVs survey at these depths. So it's, in a way, it's odd. There's less known at about 100 meters than at like 1,000 meters. And of course, there's a lot of life at this depth. I'll just zoom out a bit.
Gary, what's that behind you?
I'll zoom in. A coral. A hard coral, maybe? No. Ascidian?
Some kind of ascidian.
Ascidian.
You can see the sort of siphons on the side and lots of other little holes. Filter feeding away.
Yeah, okay. So we can grab this coral here.
I'm going to have to grab something out because I'm using that arm right now.
I should get that other ID book in here. It's got all that kind of crap in it.
Yeah.
No, I'm gonna go grab it.
Okay.
It'll help with that kind of thing.
For the camera. Camera.
I'm gonna put Ben on beer duty.
I'm gonna move over.
I got it. Okay. He's just trying to weave himself.
Yeah.
We're real close into this wall.
There's actually another coral of interest right here also, so we could stick here. It was like a black. Now we can't find it. But...
I can try to get the started on that just a little more.
You're looking for the decapod thing?
The ascidian.
Oh, the ascidian. Oh, okay.
Cool to find. You grabbing that coral?
Yep.
Nice.
There's a black coral, then there's something else of interest that we might look at. So as you can see, we could do entire 9-hour dives at this depth, just moving little by little across at this depth range. There's just so much life, so many different corals. We actually do have time, so we've got about 2.5 more hours. So we will take our time and collect and explore. So really what I've always wanted to see on this trip was a pygmy seahorse. Now that we saw it, I don't know, what's next? What are we dying for now?
Tiger shark.
Make sure to get the bottom.
Yep.
Yeah. What do you think? What's the most sought after thing that we want to see now? We saw a pygmy seahorse, which is unreal.
Yeah.
What's the number one thing?
After this,
Definitely some kind of
gosh, something large.
Something large. Well, I think the sharks and the whales, I think they stay away from the ROVs. We do want to see a turtle.
That would be cool, wouldn't it? Yeah.
But honestly, I think they stay away from the sounds and the lights of the ROVs because we never see them, but they're all over here.
Yeah.
I had an ROV get bit by a turtle in Brazil.
Really? Did it do damage?
Very basic. Go back and forth.
No, that's okay.
Where would you like this to go?
1B? What's in 1B?
We've got rocks.
1B. So there's a coral in 1A, coral in 1B, right? Yeah.
Yeah. What did you want to like this?
A kraken. Yeah, right. Okay.
So what's the kraken?
Yeah, kraken would be a good one.
Okay.
Yeah.
You can settle down, you're good.
Manta ray, sure. Yeah, all the charismatic
We saw
megafauna. Oh, we did see an octopus.
Yeah.
Yeah.
There's the second thing you wanted to get?
Yeah, once this goes in the box, 1B, please. We'll zoom out. There's like a black. This. Curious to have a zoom in on that. Every day it's like Christmas or Hanukkah, you know. Really, it's more like Hanukkah. We have multiple days of presents.
Yeah.
Christmas would be like one dive.
Advent calendar.
There's always Boxing Day.
Oh, right, okay.
The coral looks a little bit like a menorah.
It is like a menorah. You're right.
What's that called? The I9 baby coral? It's the one that looks like that?
I don't know. Yeah, I don't know.
I have a photo of it. Yeah.
The candlestick coral.
That was when we found the sea panda.
Yeah. Someone should make a menorah shaped like a coral. That'd be pretty cool.
I think you're probably the man for that joke, Jeremy.
Yeah. I'll need someone like Hanukkah to make it for us.
Out of jewelry.
Yeah.
There we go.
Yeah, that tunic.
Whoops, sorry, buddy.
Probably something like this. Little Globos ball.
I'll go with the Globos ball.
We can get them in colonies of lots of little individuals.
Alright. I'm on the ball a little bit.
That's alright.
Okay. We gotta find it again.
It might be blocking it. Yeah, it might have to.
It might be right under the arm. Or,
I mean, I know it looked like this one, but it was a different.
yeah, it was black.
Are you going to completely move the arm or just get down a little bit?
No, yeah, completely new because you're blocking it here. That's all blocking.
Got rock at 130.
There it is.
Right there.
110 is what we're after. Yeah, I think we got a 130, didn't we?
We did get a 130.
Let's get a
so we're
130.
after 110.
Let's just focus.
Oh, yeah.
geology's dominated today. So.
I'll just. Yeah. You got it. Okay. Just. Okay.
Alright.
This black. Oh, cool.
So what do you think, Jeremy?
Well, we're thinking it's a soft coral. We're communicating with Kathy McFadden now. So who's helping direct some of the soft coral collections.
It's not one of these ones that's like zoanthus that have colonised branch, is it?
I don't think so. You don't see the zoanthids, coming out.
Yeah.
So these are like little polyps that are. They're sponge on the base. Why don't we just take a grab and put it in BioBox 1B as well? And yeah, we can look at it later.
You ready, Chris? I can try to get rock about just a little bit more, give you a little bit more room.
Take it a second.
Cool.
Nice. Someone just got the wrong number for Pizza Express.
We'll get a rock here as well, right?
Joe thinks there's something a little bit further over on the right. Might be a bit cleaner.
Right.
But yeah, around this step would be good. Nothing's really obvious. It looks a bit grungy in this spot.
Covered in stuff.
Okay, yeah, sharp eyes.
Amazing, huh?
No.
Just, just kind of subtle movement behind you.
Yeah, just trying to this.
It's obvious. Yeah,
I can't.
Really difficult to see.
You gotta go a little bit more. No, I have to go out a little more.
I knew we'd find it, the feeling. They're here, right?
I feel like we've seen so much on this dive, you lose track of what dive it was, but it was all this one dive.
Yeah, every dive is magnificent. They all blend into this one ridiculous experience.
Starting with all those chimeras and the amberjack with the cookie cutter bite on its side, and then we've come up this face of gradually more and more colourful coverings on all of those amazing rocks.
Yeah.
And now we're well into sampling mode.
Super, yeah. Oh, there's so much here. It's like a buffet. Not unlike our lunch.
Amazing lunches.
Right here.
Yep.
Ah, excellent. We've got Dougal online providing some great ID.
Epizoanthus.
Epizoanthus. Oh.
So he's thinking about a zoanthid going up the stem. We saw. Okay.
Yeah, maybe. We will see.
The thing we were looking at before kind of curved down like this at the bottom, away from, you know, the white tip.
Yeah, it's down. Yeah, that's it. We got a different angle on it now. Here's this.
The second, see the other one. Oh. I got it. I say, but it had like a white.
It still does. We just got a different angle on it. That came up. We were looking.
Yeah, no, I just want to make sure I get all of it. Yeah. I see it. I see it. There's a rock on it.
I would never do that. Oh no, you were mean to me.
What?
You were mean.
I wasn't. Comment my instinct further to the next team.
Yeah, cat the second's probably a zoanthid.
We'll try.
Look at that now.
Yeah. Okay. Yep. That makes sense. A little bit like what we had yesterday, sort of growing up around the dead branch.
Yeah.
Oh, it's not on it.
Oh, rock.
Learning a lot about zoanthids on this trip.
I would move that rock.
Yeah. Come on, rock, we're on the way now.
Yeah, okay.
There we go. That's the one. Tricky angle.
No, it's just I sort of rock in front of it, and I actually think it was on the rocks.
No.
So I can see a cycle like this with those big round flat lines in the zip cam, right at the left edge of
Up to Sarah's.
I think this might be a cycle like this.
No, they're a bit more grey. They've got more of a grey colour. If anything, we've seen sponges look like that.
We're going all the way up to 60.
But yeah, we should be seeing.
Yeah, it'll be interesting.
Left to Sarah's. So they've got that more browny colour.
Yeah. I'm surprised we haven't seen them yet.
What? Leptoceras?
The Leptoceras, yeah. We saw them as deep as 150 at the very beginning of the lake too.
What time? What depth?
150.
Out in the Coral Sea.
Yeah.
What about yesterday? When did we? What was the depth?
About here, a little bit deeper than here.
Yeah. Okay, there you go.
The aspect between today and yesterday is quite different. So
Right.
yesterday we were coming up along some quite nice, almost horizontal ridges, very gentle. This is a lot more.
Joey, do you want to look at this or just
Just throw it in. It's okay.
And two again?
Yep.
Beep.
Are y'all getting that other thing you're recording?
I'm not sure if I'll have to zoom in and have a good look at it first.
Yeah, I know where it is. Alright, potentially a zoanthid being dropped in our box. Looks like we got a soft coral with it.
Bonus.
Yeah, that's right. So now we just want to look. Yeah, you know where you're going? There it is.
It looks coral-like to me. It is, yeah.
It's
Leptoceras. Yeah. Do you see the ridges?
Yeah.
Yep.
And that's central.
This one will be too hard to collect. But yeah, so
This is our first one.
we're seeing them now. So good to see.
So this is a zooxanthellate hard coral. Zooxanthellate meaning hard coral that has zooxanthellae inside it, so photosynthesizing algae in its flesh. And this is one of the deep water specialists that our colleague Tom Bridge from the Museum of Tropical Queensland is particularly interested in. And it's always great to come from below and up, and of course you find the lower depth limit, so it has this classic plate-like shape, and that's really to harvest as much light as possible. So here we are at 110 metres. It is the mesophotic zones, the twilight zone, and if we turn all the lights off you would see light coming through, but it'd be this sort of dull blue colour. And but this coral, there we go, thank you very much.
Thank you.
This is the ambient light, and of course these are laser beams down the bottom, but this is how much light is there normally, right? So everything's kind of bathed in this, it's got this blue colour to our eyes, to our human eyeballs. There we are, looking up. Thanks very much, that's great. But these deep water specialists can I guess harvest the available photosynthetic light to charge the zooxanthellae, which live symbiotically within the flesh of the coral, and you know it's a real quite amazing to think that this coral is specialised to live at these depths. And of course as we go higher we're going to see more and more different species of zooxanthellate hard coral. And we also need to get a rock from this depth. This is also around the that lower sea level depth that we've been talking about yesterday and today. So we're looking for biology and also geology. So we're just going to try and find a nice black, clear rock. Oh yeah, kind of possibly zoanthid covered branch down on in this lower centre of the screen now.
He's been doing it.
Yeah.
I just thought the saw would have done it. Guess not.
Should I point? I don't think you even need to point there.
Okay, so we're trying to pluck out a nice looking rock. Marty's just making her choice.
Just introduce us.
A very considered choice.
That's what I want to talk about.
So
Blue light is on. It's picking up all of this. If you need to stop, just turn that off. But just get in there and introduce yourself.
So we've just got Ben coming to join us in the ops room at the audio seat. You want to say hi, Ben?
Hello, everybody. It's a bit loud.
So we're in the middle of a rock collection at about 110 meters. Coming up through the mesophotic, and every 10 meters we go up, we're getting more and more coverings over the rocks, sponges, ascidians, lots of black corals,
and some
Granian fans.
A lot more photosynthetic life at this depth, isn't there?
Mm-hmm.
We've just seen our first Leptocerus coral.
Oh, right, okay.
It wasn't, it was too deep. It was a bit awkward to actually get it, but we got a good look at it. So.
I'm sure there'll be plenty of opportunities to get some samples.
Yeah, it was very small compared to, the first one we saw yesterday was like dinner plate size. This one was
Yeah.
I don't know, teacup size.
Assume they're slower growing at the slightly deeper depths.
Yeah, we got a fantastic specimen yesterday that the guys managed to get whole into the box. It's been wrapped up, preserved, ready to go to the Museum of Tropical Queensland with Jeremy.
These guys got some pretty amazing dexterous skills with these arms.
There's some crumbles on the bottom, but
There's a good knob right
there in the middle.
That was 1% as well, right?
Right here.
Seeing lots of amazing tunicates, you know, those big globose ones with kind of a honeycomb pattern and lots of
Looks like they got some
siphons on the side.
encrusting sponge as well now.
It's mind-blowing colours though when you think about how deep and how little light you've got down here.
At least with the lights on. Well, that looks like it's very loose, doesn't it?
Yeah, so it looks like there's so much covering a lot of these rocks that it's pretty tricky to make sure we're grabbing the actual rock rather than the stuff over its surface.
You would assume all the bits that are sticking out are all bio-treated, so they're going to be
So what we're trying to get is
all loose material.
The internal framework coral, the original, living skeleton, and to do that we actually have to break off a fairly big chunk. Because a lot of the outside, the surface of this limestone is bio-eroded with modern stuff, and that confuses the aging, the age issue. So we're looking for something big enough that we get a nice fresh white face.
When it's sticking up, I think that's going to be just crumbly stuff as well. I think it's going to be rock right there. Just got to get
Looks like they're...
Red.
Which one?
Round.
This. No, it's pretty...
It's hard to tell from far away.
Could be a closed-up anemone. Oh no, actually maybe that's I think it might be an echinoderm. There's some description. Like a pillow, something like a pillow star. Yeah,
it's a sand dollar type.
Yeah, it's hard to say. Or it could be another tunicate. Insectoid has said it's a marine tomato, which is
tomato.
about as good as my guess.
I like that. Yeah, some kind of urchin. It's got a bunch here.
Yeah, maybe if we get another angle on it in a second after these guys have successfully got a sample.
We've got... That's a fun accent. Where are you from, Ben? Give yourself a little introduction.
You can't
here.
guess. I'm from the United Kingdom. A long way from home right now. It's so loose, isn't it, this material? And there'll be boring sponges and things that are, like Rob said, they'll be bio-eroding, breaking down the consistency of this substrate. Yeah. Ooh, this is so tense now, I think.
Suspense is killing me.
Yeah. Oh.
Alright, let's see what we...
You have to wait for the dust to settle. I think it's just so...
It's really just being,
so loose.
cemented by all of this life, all of these things growing on these rocks. It just builds up this layer of unconsolidated, really.
Well, this is also in a, what depth are we in? 110. So it's also in a slow growing area for calcifying organisms, so it's not going to grow back. You know, stuff's not accreting at the same rate as it is higher up, so it's, but
a little smushed.
Yeah, get rid of that.
Shaking that
Start
off.
again.
I think particularly on these quite steep slopes, you've still got a lot of sediment coming off the top and just building up these layers over the top of everything. So we're going to go on a hunt, I think, for
looks like it.
Maybe something a little clearer, cleaner that we can get a nice chunk of.
Yeah, it's hard to know what's going to be the best target until you start probing it, I guess. See Jeremy eyeing up all these black corals.
Yeah, I think these round things.
Marine tomatoes, right?
The marine tomatoes may be sponges. Or maybe that's something different.
Amelia says it's nice to hear some fellow UK accents.
We get everywhere.
You can do an impression, Jeremy. Teas and crumpet, yeah, that's what it's all about. Actually, do have a cup of tea somewhere.
Oh, yeah. Yeah, beautiful crinoid there, all spotted. Yeah. Oh, just hanging on. Yeah, this is a tricky section.
John Holmes reckons we need a hydraulic jackhammer. I think that's been suggested before.
This isn't one in the pocket.
There's of course the issue of every action has an equal and opposite reaction.
Setting the ROV backwards.
Yeah.
It's essentially like being in space.
Okay, I'm going to reconsider my. I think those are sponges. Those
I
red
think those are
globular
sponges.
things.
Well, these ones are actually a bit more irregular. We've definitely seen a couple that are pretty spherical. You know.
Yeah.
Well. But yeah, I would go with sponge.
I think that would be my best guess. Best guess for now.
Push away.
Yeah, just so loose. Very loose.
Small pieces.
I almost think you could suck some of this up.
Try it.
You want to be down 310, right? Or 110. Same here.
Try that little business thing.
Kind of got everyone here in the room scouring the screen for a potential grab candidate.
There's lots of things that look like they'd be good candidates, but I guess you just don't know until you start having a tug.
I mean, this kind of thing is tough. Even when you're diving, trying to collect things, you start going at something, you're thinking it's a nice hard rock, and it turns out to just be a pile of sand or mud. Got a nice huzzah for the encrusters in the chat. It's excellent. They are the cement mixers of the reef.
There you go.
Oh,
whoa!
Get after it.
Come on.
Yeah.
We'll chase this.
Good work, J-Rod.
See it. Don't fall off.
Oh, excellent.
Beautiful.
All right. So our rolling rock sample has
So
just
from one of the other screens, we've been able to see where this
stopped on this ledge.
This nubbin has stopped.
I'm gonna go and grab it. Just wait for the dust to settle. And then I guess we'll be on our way further up.
Alright, so you want to be look down with the forecast, right? You're not warm up so much, you're on the lock right?
Yeah.
We're getting set up.
Just gonna put my porch up real quick.
Yeah. The tension is killing me.
This is a nice piece if we can get it.
No.
Alright, just parking ourselves up, making sure we get a nice clear view of the piece we worked so hard to break off up there.
It's wee business. These guys are amazing.
Alright, go ahead, Chris, you go on for it.
Here we
I
go.
don't think I've ever been so excited about a piece of rock. This is welcome to your world, Marty. Here.
It's contagious.
Contagious, yeah, it is.
It's a nice
Ooh.
big sample. Maybe even too big for our
Oh, I think it's going to be good.
calculator. Oh. So much on it, encrusting sponge.
There's some CCA. Looks like there's
Yeah, still soft, you know.
maybe you just need to scoop it. It looks like there's a coral recruit as well. I don't know what that, or I'm not sure what that is there.
Yeah, I see it. It's we'll have to fight the geologist to take a look at this when it comes back up to the ship. It looks like it's got all sorts of interesting things on it. Yeah, excellent.
Right, I'm on tenterhooks here. Oh my goodness! Wow. Oh.
Take a look at it. Put it in. Alright, you want it?
Quite something.
I can bring the porch in a touch. Can I help you?
Yeah, let's.
Just coming in slowly if you. Wow.
And this tension is killing me.
What are we going to do after this?
Yeah, keep cruising up
Yeah.
to shallower depths.
We
You know, try to get to 60 or 70 if we can.
is that what we think it flattens out?
Yeah.
Yeah.
Oh, we might flatten out at 90 anyway. Yeah.
Thank you very much.
Oh, nice one.
Good work, guys.
Oh my
It was
goodness,
well
that's intense.
earned rock.
How is it for external temperature now? Is it 23? Is that going to make it too warm for the...
No, we're fine right now.
Yeah.
Okay, so currently at 113, 114 meters, and we're going to start to, or continue our ascent up this face.
I'm assuming that's where we are.
I think as Rob was explaining earlier, the top of this spur and groove canyon system is eventually just going to come up on top of the shelf break, and it's an interesting one, or it's different to the dive we did yesterday because we don't have any emergent reef sitting.
No reef crest at the top.
Reef crest at the top. So this is completely open to the shelf. So it's going to be interesting to see when we get up to that sort of sub 100 meter level how different things are going to be. Very, very craggy. You've got. It's very crenulated.
That's exactly what I was going to say.
Outcrops and walls. So quite a different aspect to what we were getting yesterday. There's a little chromy damselfish down there. Oh, and some kind of Anthea or tilefish. Lots of lovely crinoids on those gorgonians.
Does Jeremy want more samples?
He probably will, but I think we'll keep going.
He always wants more samples.
Jeremy always wants more samples.
I guess
Yeah, look at all these structures. Just kind of the combination of the fossil coral, the life growing on top. Oh yeah. Yeah, beautiful.
Anthea.
Maybe some kind of fusilier, Cesianid.
Yeah. It's amazing everything that lives in these all these little crevices and whatnot. It's
beautiful. I would, maybe tilefish, some kind of lyre-tailed Anthea. All right.
Yeah. You got lots of these sort of sandy sediment slides that you get, sort of avalanches that you get in shallow reefs in the grooves as well. It's quite a similar looking site. Cool, and that's the 100 meter mark. Well, it's kind of serranid.
Yeah,
it's
like Plectropomus something.
Plectropomus species.
Maybe maculatus. Oh.
I feel like one of them. Yeah.
Yeah. Okay.
Yeah, I think for anyone in Australia watching this, they just call that a coral trout.
Coral trout.
Sure, so.
You gonna keep following them or keep going up?
What do you think, Jeremy?
Yeah.
Do you want to get anything else?
Yeah. Do you want?
There'll be something, but not right here.
Yeah, okay. Good.
Is that a fish?
Where?
Alex spotted it before.
I see
Looks
what
like
you-
this. They look like sponge.
No, I think
think
that's
that's,
a sponge. I think.
Yeah.
I know what you mean. They do get
Sponge.
frogfish that camouflage up there.
Yeah, I know where it is.
Okay.
Are we going to try and find you want to try and find Acropora?
If possible.
Yeah,
I feel
okay.
like this might be a place where we can find it.
Going to get my Acropora eyes on them.
Yeah.
Look at this sea fan, black coral sea fan.
Probably still a little deep, aren't we? Yeah, but
Yeah, we'll head up a little bit shallower. We still have a few hours, don't we?
yeah.
We don't want to get up too quick.
Yeah, not 2 hours.
Leopardus.
Okay.
Yeah. So,
Electroponus.
good old-fashioned coral
Good
trout.
old-fashioned coral trout.
And what's the shallowest we can go here?
Well, up to these guys. I mean, 60 is where the transect should go to.
Okay. So an hour between
60.
180 and an hour between 80 and 60?
Give or take.
Pretty good spot right now.
Pretty good. Yeah.
Pretty good spot.
Okay.
Okay. So
Maybe
first,
just back up a bit less than that on each zone.
45. Yeah.
So keeping our eyes out now for some of that Leptocereus, the hard
Yep.
coral we've been trying to collect on this
I think that's a sponge.
expedition. Is that one? Small one?
Maybe.
Yeah.
Yeah, it's tiny.
It's gonna be hard to collect. It's
Yeah.
gotta be coming off the substrate.
So you can get the
Yeah.
manipulator underneath here.
Big Argonian sea fans.
Huge, yeah, beautiful. Oh, it's starting to flatten out a little up here.
Well, we've got these sort of ledges that create a sort of near horizontal aspect, which can really make a difference for how things grow and where things can grow. So it's a pretty interesting feature of these. What can look like just a sheer wall, you've actually got quite a few ledges and bluffs that present these nice horizontal flat areas.
Yeah, some of it, you're getting into like nesting habitat for some species of fishes. Yeah.
Jeremy has pygmy seagulls written all over it.
Yeah, that's quite a find earlier.
It looks like that orangey.
It does, doesn't it?
Is that a nudibranch in them?
It's potential. Do you want to zoom in? Yeah.
So this is the
You
zoom.
can take the con.
Alright, something interesting. Looks like it's like epifaunal there. It's growing into the.
We saw, and we can see some bare patches here on this Gorgonian, like we had before. Maybe some grazing.
Predating on the. Oh, it's a
Yeah.
clam. It's a bivalve here.
Oh, okay.
Yeah.
Yeah.
Well, I'm still looking for seahorses.
Yeah, everyone
Yeah.
get your seahorse eyes on. Alright, it took me ages to home in on that one that you guys saw earlier.
Yeah, it was Joe Rod who picked it out.
Quite fine.
And your eyes. You feel it's got to be a.
This is very interesting bivalve. It's clamped.
Yeah.
It's clamped around the branch.
You wonder how they can settle on
We
something
tend to
like
find on fans like this.
But once they're dead,
bivalves that kind of.
once they're dead though, or on a dead section, or this.
Dougal earlier, but similar
Yeah, comb oyster. Yeah.
mouth opening, very jagged.
Ooh.
Ah, yeah.
Some kind of comb oyster, we think.
Cock's comb oyster, or
Oh wow, look at that.
some
About to
great
head into the sun. So it goes up quite
upward,
shallow.
And
I feel like we're getting into Acropora country here now.
oh, we are. We still haven't collected one yet.
keep those eyes peeled.
Yeah. I feel like looking down the wall is probably the way to spot them. You know, you see that characteristic.
Gorgonian whip.
Guessing the oh no, just the aquapora plates.
He thinks you guys aren't good enough.
I didn't say that.
Yeah.
I said I think it looks different.
That
It
I
looks,
picked up the other day?
No. It's a branching hard coral, like a
stagger one.
plate,
Yeah.
and then it'll branch up.
We can do it.
They can do anything.
Looks like a tunicate on there.
I want to know.
Beautiful outside.
Yeah, on the left. Yeah.
Yeah. Some grazing
Some predation or maybe
going on.
disease.
Bought a few more fish and piers and a little hogfish down there. More of our chromies, some damselfish.
This weird thing. I want to figure out what that is.
Have you got one yet?
You got one of those, didn't you?
On this dive?
This thing that we collected earlier, that black thing.
Yeah.
Regardless of
Not interesting. I'm pretty sure you get them there.
Oh, wait a second.
Sample that thing?
If you can get the whole thing, then yeah. If you think it'll be too hard, we'll wait for the right one to grab.
There's a lot of these really nice coral right here.
Yeah, I think just there's going to be a better candidate, isn't there?
You okay?
We'll just make a note that we saw that at 87 metres and
Right in the middle.
good note taking, Jeremy.
Yeah, you're going to use starboard or port or starboard?
It's out right there.
There are a lot of fans in the way, aren't there?
They are.
Yeah, this is quite an overhang here. It's
Needs to get out of the way.
yeah.
Oh, that thing. That's a good look. Serious.
I want to see my fish. Long gone. Oh no, it's still there. Very near the coral.
It'll be gone in a second.
Oh, some kind of
Yeah. And T-Series starting to grow behind. What's that? Yeah.
It could be a black coral, or it could be a bryozoan.
Oh. Lots of Halimeda gravel.
I said it's tricky.
have you?
No, not yet. Ooh. I think it's
Yeah, I think it's
chance.
other stuff. I just don't know.
Cirrolabris. Yeah. Oh, no. Getting ready. Consistency of a potato chip.
B is pretty full. You can put it in C.
Seems to be still slight.
Now that you're there, you can maybe try for
So it looks like we had a little bit of connection issues just now from the ship. So we're just tuning back in. We are in the middle of a quite tense collection of this really nice Leptocerus specimen. You guys are doing an amazing job
My heart was in my mouth. It's amazing.
getting this into the box. There we go.
Oh, amazing work, guys.
Going
I
around
wasn't
the
even
room
doing
now.
anything and I was still there. A little bit longer at 8.
Spot.
5 o'clock? Did y'all say 5 o'clock or? Yeah, you know, on deck.
Off bottom at 5, so basically 10 past.
A green.
Yeah, there's 2 of those sponges back to back.
Is that a shrimp J-rod or a seahorse? Take a look. Okay.
Yeah, so this is where like
One of those ledges.
yeah, look at that. Looking for aquaphora. You think we can manage an app? Yeah, keep the temperature low.
Where's this foot?
Channel in between 2 of the main Ribbon Reef crests.
Sponges around.